The handouts or review materials must be treated with utmost confidentiality. It shall be the responsibility of the person whose name appears therein that the handouts or review materials are not photocopied or in any way reproduced, shared, or lent to any person or disposed in any manner. Any handouts or review materials found in the possession of another person whose name does not appear therein shall be prima facie evidence of violation of RA 8293. Top-notch review materials are updated every 6 months based on the current trends and feedback. Please buy all recommended review books and other materials.
We will now go to metabolism. Ta-da-da-dan. So I'll give you first an overview of metabolism, and then we'll go to the carbohydrate pathways. So metabolism is the sum of all the chemical reactions in a cell, tissue, or the whole body. That's why you will hear cell metabolism, musculoskeletal metabolism, nervous system metabolism, or human metabolism. Now you have to remember pathways that make up metabolism as a whole. They can either be catabolic or anabolic. Our mnemonic for this is A, B, C, D. So anabolic build, catabolic, destroy, or degrade. So for example, when we say anabolic pathways, these pathways synthesize compounds from smaller raw materials. These pathways are endergonic and divergent. So for example, from glucose, we can create glycogen. From amino acids, we can create proteins. So these pathways are endergonic. So if for endergonic, what's the value of delta G? Delta G is positive. And therefore, these reactions require input of energy. They're not spontaneous and ordinarily not favorable. When we say divergent, from the same raw materials, we will be able to produce different products. So for example, glucose, we can use it to produce glycolipids, glycoproteins, polysaccharides. Examples are protein and triglyceride synthesis and glycogenesis. So glycogenesis meaning synthesis of glycogen, triglyceride synthesis, and as I've said, protein synthesis from amino acids. How about catabolic pathways? Catabolic pathways require breakdown of larger molecules. So kabaliktaran, degrade or destroy. They are usually oxidative reactions. So they release energy. If you break down larger molecules, you release energy. That's why they're exergonic and convergent processes. And when they release energy, they produce ATP, mainly via the respiratory chain. So electron transport chain and oxidative phosphorylation. Examples, glycolysis, the breakdown of glucose. Beta oxidation, the breakdown of fatty acids. And glycogenolysis, the breakdown of glycogen. So dahil exergonic, the value of delta G is negative. Spontaneous? Yes. Favorable? Yes. These pathways are convergent. So if you look here, when we digest, so when you digest carbohydrates, proteins, and fats, so your carbohydrates, we break them down into monosaccharides. Your proteins, we break them down into amino acids. Your fats, we break them down into fatty acids and glycerol, yung triglyceride. You will see they will converge into one central molecule, acetyl-CoA. So acetyl-CoA, that's a central molecule of the metabolic pathways. The digestion of carbohydrates, proteins, and fats converge at the point of acetyl-CoA. Why? Kasi si acetyl-CoA, siya lang yung papasok sa citric acid cycle or Krebs cycle, which we will discuss later, which produces a lot of ATP for the human body. So remember, catabolic pathways are convergent, and they converge into one central important molecule, acetyl-CoA. Now, meron tayong tinatawag na amphibolic pathway. When we say amphibolic, it is both anabolic and catabolic. Nag-iisa lang yon sa biochemistry. Ito rin laging binabanggit. It's your Krebs cycle or your citric acid cycle. It serves as a link between the anabolic and catabolic pathways. Meaning to say, it doesn't matter whether you're well-fed or fasting, whether in an anabolic state or catabolic state, tuloy-tuloy lang siya. So that's the Krebs cycle or citric acid cycle. And again, the molecule that enters the Krebs cycle is acetyl-CoA. How do we regulate metabolism? Kasi 'di ba, sa dami-dami ng pathways natin, how do we know which one do we need to activate? Aling pathways dapat yung naka-on? Aling pathways dapat yung naka-off? Paano natin nasa-switch on? For example, yung specific pathways if we're well-fed. Paano naman natin nasa-switch off yung iba? What if we go in a fasting state? Ano naman yung masa-switch on? Ano naman yung masa-switch off? Ang galing naman ng body natin, no? Kasi ibig sabihin, lahat sila nakasynchronize. Kunyari, paano malalaman ni heart, no? Well-fed na tayo ngayon. Dapat si stomach, well-fed state din. Hindi pwedeng si heart, ang mindset niya, well-fed, si stomach, naka-fasting. No? So, kailangan, paano nangyayari na sabay-sabay sila? So, in physiology, I'm sure you also discuss this. You have your second messenger systems. So, your second messenger systems, what's their role? No? Their role, okay? Their role will be to make sure, no, that the message from the hormones or from the neurotransmitters, they're carried out in the end organs. Okay, so for example, okay, pupunta tayo kay insulin tsaka si glucagon. So let me introduce you to the concept of the well-fed and the fasting state. Si insulin, it's a hormone that we produce when we are well-fed, di ba? So ang role ni insulin, kailangan masabihan niya lahat ng tissues, lahat ng organs sa body natin, uy, we are in a well-fed state. So, meron siyang ngayong mga pathways na isi-switch on, meron siyang mga pathways na isi-switch off. In contrast, si glucagon naman, it's the hormone of the fasting state. Meaning to say, if we are on a fasting state, so which is 4 hours after a meal, tataas si glucagon, and i-inform niya lahat ng organs and tissues, okay, fasting tayo. Ito yung mga pathways na kailangan naka-switch on, ito yung mga pathways na naka-switch off. Tick, 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 tick. Okay. Wala dapat pasaway. Dapat pag sinabing well-fed, well-fed lahat. Pag sinabing fasting, fasting lahat. So how does insulin accomplish that? How does glucagon accomplish that? So si insulin, if you look at the table, it has a second messenger, tyrosine kinase. So you see in the table, tyrosine kinase is the second messenger of insulin and insulin-like growth factor. So si insulin, yung message niya para masabi niya dun sa cell, magre-rely siya kay tyrosine kinase to amplify the message. Okay? Para masabi niya yung message so doon cell. Bakit ganoon, no? Bakit kailangan pa ni insulin, no? So let me illustrate that to you. So for example, let me just do this. Okay? So kunyari ito si cell. Okay? So that's the cell. So si insulin, okay? So ito si insulin. Okay? So, actually, wag na lang si insulin. Mas maganda palang gawing example si glucagon. Okay? Kunyari si glucagon. So, si glucagon, meron kasi siyang message. Diba? Ano yung message ni glucagon? Okay? So, yan si glucagon. Ang message niya ay okay, yan yung message niya. Sabi niya, fasting tayo. Okay? Pero mahina lang yung boses niya kasi mag-isa lang siya, di ba? So, ano sabi ni glucagon? Fasting tayo. Okay. Hindi pwedeng ganoon kasi kailangan masabihan niya lahat ng tissues, lahat ng enzymes, lahat ng body, buong body. Kailangan masabi niya, fasting tayo. So, anong gagawin ni glucagon, no? So, si glucagon, okay, didikit siya dun sa membrane. Dun sa membrane, meron diyang receptor, no? Meron diyang receptor. Merong receptor si glucagon. Tapos, ibubulong niya yung message niya. No? Pagbulong niya dyan, no? Pagdikit niya dyan, ibubulong niya yung message niya. Tapos, ano mangyayari? Yung message niya, mapapasok sa loob. Nandun yung second messenger, si cyclic AMP. No? Inside the cell, ATP will be converted to cyclic AMP. At itong si cyclic AMP, bubulungan siya ni glucagon, fasting tayo. Anong gagawin ni glucagon? Anong gagawin ni cyclic AMP? Si cyclic AMP ngayon, siya ngayon yung magdi-disseminate sa mga different organelles. Kay nucleus, kay mitochondria, kay ribosomes, no? Kay Golgi complex. Lahat yan sasabihin niya, mag-iikot siya sa buong cell. Sabi niya, fasting tayo, fasting tayo, fasting tayo. So, para lahat ngayon sila alam na, okay? Alam lahat na fasting tayo. Okay? Alam lahat na fasting tayo. So, anong naging role ng second messenger? Yung second messenger, anong ginawa niya? Tinransmit niya yung message ni glucagon. So, si first messenger na fasting, tinransmit niya intracellularly. So, from outside, na-transmit yung message sa loob. And what else? The message was amplified. No, meaning to say, no, nasabihan lahat. At yun ang gusto natin. Yun yung gusto natin. So you will see here, no? So si glucagon, ang second messenger niya, no, si cyclic AMP. No? So cyclic AMP, what does it do? Cyclic AMP amplifies the message of glucagon. And that's important. Okay? Now, sino pa yung mga ibang mga second messenger? So, sabi natin kanina, si insulin ang second messenger naman niya, si tyrosine kinase. So, didikit siya dun sa receptor, tyrosine kinase will transmit the message to the inside the cell para lahat ng enzymes, lahat ng organelles alam, uy, well-fed tayo. Now, who are the other second messengers? So, your epinephrine and norepinephrine, no, of the sympathetic nervous system, they rely on, you will see magkakaiba sila depending on the receptor. Pag alpha-1 receptors, they use IP3DAG. Pag beta-1, beta-2, and alpha-2, cyclic AMP naman yon. Nitric oxide and ANP, the vasodilators, they require cyclic GMP. What about your steroids? Your steroids, they do not require a receptor on the cell surface. Why? Because your steroids, they're hydrophobic. They're non-polar. They will be able to cross the cell membrane. Diretso na sila sa nucleus. Kasi nga, hydrophobic, non-polar sila. Palulusutin sila ni cell membrane. And therefore, hindi nila kailangan ng receptor. Diretso na sila. So, when you look here, so again, ito yung sinasabi natin kanina, like glucagon. Glucagon will bind to its receptor. Sabi niya, fasting tayo. And then, inside the cell, ATP will be converted to cyclic AMP using adenylyl cyclase. So, adenylyl cyclase converts ATP to cyclic AMP. Yung cyclic AMP, siya yung second messenger inside the cell. Sasabihan niya ngayon yung mga organelles, fasting tayo, fasting tayo, fasting tayo. Okay. Ano ngayon mangyayari? You will see here, cyclic AMP will activate protein kinase A. And what will protein kinase A do? Protein kinase A will cause phosphorylation of enzymes. So, yung mga enzymes phosphorylate sila. Yung mga enzymes, no, pag na-phosphorylate, no, yung phosphate para yung switch. Some enzymes will be switched on, some enzymes will be switched off. So, later I will tell you how to remember it easily. But for now, tatandaan nyo lang ang end effect. Pag na-activate pag tumaas yung cyclic AMP inside the cell, ma-activate si protein kinase A, it will cause phosphorylation of enzymes, causing them to switch off or switch on. Depende kung anong kailangan natin pag fasting. Okay? How about for IP3DAG? Ito naman yung nangyayari sa IP3DAG. Very similar to that. Okay? Once for example, epinephrine binds its receptor, so alpha-1 receptor, phosphatidylinositol is converted to inositol triphosphate and diacylglycerol inside the cell. So, dun sa cell membrane, yung phosphatidylinositol, mako-convert into IP3 and DAG. Si IP3, it will cause release of calcium. Si DAG naman, diacylglycerol, will cause activation of protein kinase C. So, once again, phosphorylate yung mga enzymes because it's a kinase. And then, si calcium naman, bakit siya tinawag na protein kinase C? Kasi ang kailangan ni protein kinase C, calcium. Saan manggagaling yung calcium? Nire-release yung calcium in response to inositol triphosphate. Okay, so again, anong role ng second messengers? Ang second messengers, ginagamit natin sila to amplify the message, to transmit the message of the hormone or the neurotransmitter intracellularly. Kanya-kanya silang second messengers, and you need to memorize who is the second messenger of which. Let us now discuss the metabolism of carbohydrates. So first, let's review your carbohydrates. So remember, these are your carbohydrates, and we classify them based on the number of carbons and their functional group. The building blocks of your carbohydrates are monosaccharides. No? So, pag 1 sugar unit, that's a monosaccharide. Pag 2 sugar units, that's a disaccharide. No? And then, kapag 3 to 10, no? Oligosaccharide, more than 10, polysaccharides na yun. So here, you will see, let's review your monosaccharides. Here on the left side, you will see you have your aldoses, and then here on the right side you have your ketoses. So when we say aldoses, the functional group is an aldose. When we say ketoses, the functional group is a ketone group, no? C double bond O. Pag aldose, C double bond OH. The most important monosaccharide, of course, is glucose. No, glucose contains how many carbons? 6. So that's why it's what we call a hexose. No? So kaya sa hexose, glucose is a hexose and an aldose. So you see glucose right there. Okay? In contrast, pag ribose naman, ribose has 5 carbons. That's why we call it a pentose. No, it's a pentose, only 5 carbons. So the most important one. So again, when you look here, when you look at glucose, no? So when you look at glucose, you will see that the functional group is an aldehyde, C double bond OH. See glucose, an aldehyde, we can oxidize it. So we can add more oxygen. So when you oxidize glucose, it becomes glucuronic acid. So ulit, if you oxidize glucose, glucose will be converted to a glucuronic acid. Kapag naman natin, ni-reduce, no? Tinanggalan naman natin ng oxygen. Ang tawag naman doon ay, no, so reduction. So glucose can be reduced to its alcohol form. So the sugar alcohol is sorbitol. Okay, so remember that. If you convert your sugar to an acidic sugar, that's oxidation. So glucose magiging glucuronic acid. If you convert your sugar to an alcohol, that's reduction. So yung glucose magiging sorbitol. Anong ibang examples? Galactose. If you oxidize galactose, no? If you oxidize galactose, it becomes galacturonic acid. If you reduce it, it becomes galactitol. Mannose, if you reduce it, it becomes mannitol. Okay, so tatandaan nyo lang yun, no? Huwag nyo kakalimutan. Mannitol, of course, is used for raise intracranial pressure. You give it to reduce cerebral edema. The sugar alcohols, you see, their sorbitol and mannitol, they're very important because they attract water. Babalikan natin yung concept na yan. No? So later, remember that, no? Your sugar alcohols, they will attract water. Glucuronic acid, we will discuss glucuronic acid later when we go to your glycosaminoglycans, your complex molecules. And then finally, I wanted to emphasize this, ribose. No? Unlike glucose, ribose is a pentose. It only consists of 5 carbons. No? So see, ribose, it's important because from ribose, this is the sugar of your ribonucleic acid. And then if you remove the hydroxyl group here, it becomes deoxyribose. Yun naman yung sugar ng deoxyribonucleic acid. So that's RNA and DNA respectively. If you combine one monosaccharide with another monosaccharide, you get your disaccharides. Disaccharides are also important in our diet. No? So we have sucrose, which is the combination of glucose and fructose. And then lactose, which is the combination of glucose and galactose. And finally, maltose, which is 2 glucose molecules joined together. Review, Dr. Rubio's lecture, the monosaccharide units are joined together by glycosidic bonds. Okay, glycosidic bonds. So now, let's go straight to the pathways. No? Let's go straight to the pathways. So how do we absorb carbohydrates? So lahat ng kinakain nating carbohydrates, yung mga starch, no? Lahat yan, madadigest natin. Okay? And they must be, so review just review the digestion of carbohydrates. Remember, when you digest carbohydrates, they're eventually absorbed into the duodenum and jejunum as simple sugars. Okay? As monosaccharides. We need to break them down. Okay? So the most important will be glucose. So carbohydrates are broken down in glucose. We absorb them in the intestines. And then from the intestines, no, from the small intestines, where do they go? They travel via the portal vein going to the liver. And then from the liver, where will they go? From the liver, they will go to the hepatic vein, inferior vena cava, going to the heart, and then the heart will now pump the glucose to the rest of the body. Okay, the heart will now, so that's why, no, from the blood, it will now go to your different organs. So how will glucose enter the different tissues? So for example, paano siya pupunta sa brain? Paano siya pupunta sa muscle? Paano siya pupunta sa heart? Sa kidney? Okay? So you must remember this. Okay, let's review a basic concept. Sa chemistry, laging sinasabi, like dissolves like. So meaning to say, kung ako, polar substance ako, palulusutin ba ako ni polar membrane? Yes. No? Will I dissolve in water? Yes, because water is polar. I'm polar. But what if I am non-polar? If I am non-polar, makakapag-swimming ba ako sa water? No, ayaw ko ng water, I'll be hydrophobic. Sino magpapalusot sa akin? Yung non-polar membrane. So yung cell membrane, palulusutin ako nun. So let's try to think about this. Glucose. Is glucose a polar molecule or a non-polar molecule? Because it contains oxygen and hydrogen, no? Charged, no? So meron tayong na may polarity yan. So glucose is a polar molecule. Therefore, that's why it dissolves in blood, no? Because blood is very polar. However, will it be able to cross your cell membrane on its own? Makakapag-diffuse ba siya sa cell membrane on its own? The answer is no. No? The glucose will not be able to cross the cell membrane on its own. It will rely on what we call glucose transporters. And glucose transporters, they facilitate the transport of glucose into the cells. No? So in adipose and muscle tissue, for that to happen, you need insulin. However, for red blood cells, intestinal mucosa, brain, and liver, that's not needed. So I will illustrate this later. Just remember, papasok siya inside the cell. And inside the cell, what will happen? It will now undergo glycolysis. So glycolysis. Lysis meaning breakdown of glucose. So as I've said earlier, so you will see here, no? So if you look at this illustration, this is glucose. Hindi siya palulusutin ni cell membrane. You must remember the cell membrane is hydrophobic. It's non-polar. So si glucose, para makapasok siya sa cytosol, sa loob ng cell. So this is the blood. Remember, blood is polar. Kaya pwedeng palutang-lutang lang siya doon. Pero para makalusot siya papunta sa cytoplasm, which is again mostly water, it needs to pass through a transport molecule. So that's what we call a glucose transporter. So in here, papasok si glucose, nasa loob na siya. What do you need to remember? There are different transporters depending on the organ. So in this table, you will see the different transporters, GLUT1, GLUT2, GLUT3, GLUT4, GLUT5, and SGLT1. So, look, naka-highlight si GLUT4. So, bakit naka-highlight si GLUT4? You will see GLUT4, it's found in heart and skeletal muscle in adipose tissue. So, fat and muscle. Naka-highlight si GLUT4 because it is unique. It is the only one among the GLUT transporters that requires insulin. So without insulin, it will not allow entry of glucose into the cell. So when you look here, so this is an example of muscle or adipose tissue. So this one, so look, so merong insulin receptor. So ito yung si cell, no? Kailangan meron munang insulin. Insulin must bind to its receptor. Pag bind niya sa receptor, si glucose transporter, tsaka lang siya pupunta dun sa cell membrane para makapasok si glucose. Okay? So that's GLUT4. So bago makapasok, no, inside the cell, it requires insulin. Compare it with GLUT1 and GLUT3. So GLUT1 and GLUT3, they're found in the brain, no? So the brain cells. So yung neurons ba natin, bago ba makapasok si glucose, kailangan ba niya ng insulin? No, no. So kailangan tuloy-tuloy lang lagi yung glucose in the brain, no. So even without insulin, glucose is able to enter your neurons. Okay? How about GLUT2? No? Saan naman natin nakikita si GLUT2? GLUT2 is important for the liver and the pancreas. So, bakit siya important sa pancreas? So, here, ano bang role ni pancreas in glucose metabolism? Remember that pancreas, no? Your pancreas is the one that produces insulin. Okay? Is the one. So, here, so, ano nangyayari? Pag bagong kain ka, no? Sa pagbagong kain ka, you will see this is a pancreatic cell. Si glucose, papasok siya using GLUT2 inside the pancreatic cell. So, malalaman ni pancreas, uy, bagong kain siya. At pag nangyari yun, pag nakapasok ngayon yung glucose, look what happens? Your pancreas will now secrete insulin. The pancreas will now secrete insulin. So, kaya dapat, no? Kaya dapat si pancreatic cell, no? Hindi niya kailangan dapat ng insulin kasi siya ngayon magpo-produce ng insulin. So, liver also utilizes GLUT2. No? Your liver also, nandun din yung GLUT2. Now, what about the intestines? No? So, ano naman yung role? No? So, you will see here, GLUT2, GLUT5, and SGLT1. So, babalik tayo dun sa absorption of glucose, galactose, and fructose. So, review lang to ulit. So, if we review, no? So, let's try to see. So, for example, if this is your intestinal cell, okay? So, if this is your intestinal cell, no? So, ito yung lumen. Okay? Andyan yung lumen. No? Ito yung mga intestinal cell, enterocytes. Okay? So, remember, glucose, galactose, and fructose. So, meron tayong apical side and the basement membrane side. No? May dalawang set of transporters dyan. So, tatandaan nyo lagi sa lumen, 5 letters, no? Ang nandyan si GLUT5. Sa basement membrane, ang nandun si GLUT2. Basement membrane, 2 words. Okay? So, for example, meron kang monosaccharide like fructose. So, si fructose, dadaan siya kay GLUT5 para makapasok sa lumen. At sa loob ng cell, dadaan siya kay basement membrane, GLUT2 naman, para ma-absorb siya. It's different for glucose. Kasi si glucose, saan siya dumadaan? Hindi siya dumadaan kay GLUT5. Meron tayong tinatawag na SGLT, Sodium Glucose Luminal Transporter. Why? No? So, babalikan nyo to. No? I'm sure na lecture to sa inyo. No? Your glucose, para makapasok siya dyan, it is co-transported with sodium. And it is a secondary active transport. So, si fructose, facilitated diffusion lang siya. Pero si glucose, kailangan niyang secondary active transport using SGLT1. Pag nasa loob na siya, again, dadaan na naman siya kay GLUT2 sa basement membrane para ma-absorb into the systemic circulation. Okay? Again, so sa intestine, sa intestinal cells, that's why you will see here, no? Okay, meron tayong different transporters. So here you have different transporters. So GLUT2, GLUT5, and SGLT1. Nasaan ang GLUT2? 2 words, basement membrane. So dun, no? So from the cell para ma-absorb siya into the systemic circulation. Nasaan ang GLUT5? Nasa lumen. SGLT1, okay, it's important to absorb glucose from the lumen. It's sodium dependent, secondary active transport. So kaya nga babalikan din natin to later between fructose and glucose. Sino mas madaling ma-absorb? Mas madaling ma-absorb si glucose because it doesn't require ATP. No? Kasi si fructose, facilitated diffusion lang siya. Si glucose, secondary active transport. Pero pagdating sa basement membrane, parehas na silang facilitated diffusion. So once again, this is the muscle and adipose tissue. They have GLUT4. No, GLUT4 requires insulin to allow entry of glucose. Whereas this is your pancreatic cell, which uses GLUT2, no, to allow entry of glucose, prompting it to secrete insulin. So aside from the pancreas, where else do you find GLUT2? In the liver and in the intestines. Specifically, where in the intestines? Two words, in the basement membrane. And here, what you see here, so your brain and your adipose tissue. So you will see brain, it will contain GLUT1 and GLUT3, mainly GLUT3. Will it require insulin? No, doesn't require insulin. Papapasukin lang niya si glucose. No? In the liver, what does it require? No? Again, it doesn't require insulin. It uses GLUT2, no, para makapasok, no, si glucose. Finally, GLUT4, you see here they have GLUT4. Okay? GLUT4 in the skeletal muscle and adipose tissue, they are the ones that require insulin. So when you come to think about it, what happens in diabetes mellitus? Pag meron kang type 1 diabetes mellitus, in type 1 DM, there's autoimmune destruction of your pancreatic islet cells, right? So wala tayong insulin. So ano mangyayari pag kumain sila, yung patient na may type 1 diabetes? When they eat, no, the glucose, where will it go? Will it be able to enter the brain? Yes, kasi si glucose, dadaan sa GLUT1 or GLUT3. Makakapasok sila sa brain. Will it be able to enter the pancreas and liver? Yes, makakapasok pa din sila. Pero sa pancreas, walang napoproduce na insulin. But what about the muscle and adipose tissue? If you have type 1 diabetes, dahil wala ngang insulin, GLUT4 will not be able to allow entry of glucose. No, hindi makakapasok yung glucose inside adipose tissue and muscle. So what happens to glucose? They will remain in the blood. So yung mga glucose molecules, dahil hindi sila makapasok sa muscle and adipose tissue, nasa blood lang sila. Kaya nga meron kayong hyperglycemia. Anong kailangan natin gawin? Then we give the insulin shot. Pag nagbigay tayo ng insulin, no, external, no, insulin, exogenous insulin, papasok na ngayon sa muscle at adipose tissue yung glucose. Is that clear? It's time to start discussing the carbohydrate pathways. And the way we discuss carbohydrate pathways is by pairs. No? So, by pairs tayo mag-discuss. And usually, these are opposite pathways. And we begin with the 4 Gs. No? So, what are the 4 Gs? Glycolysis and gluconeogenesis, glycogenesis, and glycogenolysis. Again, the first 4 pathways are glycolysis and gluconeogenesis, glycogenesis, and glycogenolysis. Mas madali kasing maintindihan yung relationships nila, no? If we think of them this way. So let's analyze the term, no? So when we say glycolysis, ano ibig sabihin nun? Breakdown of glucose. So pag sinabi natin glycolysis, bini-breakdown natin yung glucose. Ibig sabihin, marami tayong glucose, kailangan natin siyang i-breakdown. How about gluconeogenesis? Pag sinabi naman natin gluconeogenesis, it means genesis, create. Neo, new. Glucose. Creation of new glucose. Isipin natin, bakit naman natin gustong mag-create ng new glucose? Ibig sabihin, wala tayong glucose. Okay? That's why, if you think about glycolysis and gluconeogenesis as opposite pathways, no? Kailan kaya tayo mag-glycolysis? Kailan kaya tayo mag-gluconeogenesis? How about the next 2 pathways? Glycogenesis and glycogenolysis. When we say glycogenesis, it means creation of glycogen. Pag sinabi naman natin glycogenolysis, breakdown of glycogen. Ano ba yung glycogen? Anong klaseng carbohydrate ba siya? Kailan tayo mag-create ng glycogen? Kailan tayo mag-breakdown ng glycogen? That is what I will be discussing over the next few minutes. But before we go to these pathways, you must always ask these questions. So pag nag-aaral kayo ng pathways, no, there are 5 questions for every pathway you will study. You need to know the answers to these 5 questions. No? Ganito lang yun, no? Tatandaan na kailangan pag may pathway kang inaaral, dapat alam mo yung sagot sa 5 tanong na ito. First, what is it for? Para saan ba yung pathway na yan? No? Halimbawa, if I ask you, what is glycolysis for? No? Gusto ko, pag sinagot mo yung tanong na yan, you don't give the definition in the textbook. Instead, sasabihin mo, yung glycolysis, Yun yung ginagawa natin pag bagong kain tayo, yung glucose, gagawin natin siyang pyruvate or lactate, depende kung meron tayong mitochondria at oxygen or wala. So, i-explain mo siya sa sarili mo in your own words. Kasi yun yung yun yung test kung naiintindihan mo talaga siya. No? So, when you say, what is it for? Dapat kaya mo siyang i-explain in your own words. No? So, kailan mo siya gagawin? No? Bakit siya important? The second question is, where does it occur? Now, when we say, where does it occur? 2 levels yan. Which organ and what part of the cell? So, when we say which organ, does it happen in the liver, in the muscle, in the adipose tissue, in the brain? And then when we say which part of the cell, will it happen in the mitochondria, in the cytosol, in the smooth endoplasmic reticulum, in the ribosomes? Okay. Question number 3. What are the substrates and the products? So what's the starting point? What's the end point? So bakit important yun? Pag alam natin yung starting point tsaka end point, maiintindihan na rin natin yung regulation. No? Maiintindihan na rin natin yung regulation. So, basic principle in biochemistry, no? In chemistry in general. Substrate, product. Pag maraming substrate, anong gusto mong gawin sa pathway? Gusto mo ba siyang i-activate or inhibit? No? Marami kang raw materials, no? Kung maraming raw materials, anong gusto mong gawin sa pathway? I-activate, di ba? I-activate mo yung pathway kasi sayang yung raw materials. Okay? Pero paano naman pag marami yung product, anong gusto mong gawin sa pathway? Pag marami naman yung product, yung pathway, sabi mo, tigil ka muna. Huwag ka muna mag-produce ng fat. Huwag ka muna gumawa ng glycogen. Kasi marami pa tayong fat. Marami pa tayong glycogen, di ba? So you will now inhibit the pathway. Which is why you need to remember what are the substrates and what are the products. So for every pathway yon. Kasi iisipin mo lang, mataas ba yung substrate, mataas ba yung product. Kasi pag mataas yung substrate, you will activate that pathway. Pag mataas yung product, you will stop or inhibit that pathway. More on that later. Question number 4. Which step is rate limiting? So, remember this. You don't need to memorize all enzymes. Pero you need to remember the rate limiting step. Meaning to say, kunyari glycolysis. Glycolysis has 10 steps. The rate limiting step is the one that's very important. Siya yung merong, kumbaga sa kanya nakasalala yung buong pathway. Okay? Sa kanya nakadepende gaano kabilis, gaano kabagal yung pathway. Bakit important tandaan yon? For example, I'm developing a drug. Gusto ko mag-develop ng drug na magde-decrease ng cholesterol synthesis in the body. Ay, kunyari, 100 enzymes yon. Paano ko malalaman aling drug yung ita-target ko dun sa 100 enzyme? Anong enzyme yung ita-target ko? Of course, I will target the enzyme that's rate limiting. Kasi yun yung magiging pinakamalaking impact overall in decreasing the synthesis of cholesterol. Okay? And finally, what are the diseases associated with the pathway? So there are times when they ask a lot of clinical questions. In fact, mas important pa than memorizing the enzymes, the substrates, will be the clinical correlates. And there have been instances when they would ask cases. So they would give the case before asking about the chemical pathway. So again, always remember, what is it for? Where does it occur? What are the substrates and products? What's the rate limiting step? And what are the diseases? Most probably, you should not memorize all enzymes. Kahit ako, hindi ko kabisado lahat ng enzymes. Okay? Kahit ako, hindi ko kabisado lahat ng enzymes. Hindi ko kabisado lahat ng substrates. But, I will tell you which are the most important ones and which ones you should know by heart. Don't focus so much on the details. As I've said, especially if you're watching this for the first time, your goal is to understand. Okay? Kailangan lang ma-explain mo sa sarili mo in your own words. Para saan ba yung pathway na yan? Bakit ko ba yan kailangan? Anong mangyayari pag binlock ko tong pathway na to? Anong mangyayari kapag hindi to gumagana? Anong disease yung makukuha ko? How will I treat that disease? Okay? So let's begin with glycolysis. So glycolysis. So again, glycolysis is the breakdown of glucose. So lysis plus glyco. What is it for? This is the major pathway for glucose metabolism that converts glucose into 3 carbon compounds to provide energy. So naalala nyo kanina, di ba? Yung glucose, no? After we absorb them in the intestines, they are distributed to the different organs or tissues, and to be able to enter the organs and tissues, they require glucose transporters or GLUTs. And once inside the cell, glucose will undergo glycolysis. So where does it occur? It occurs in the cytosol of all mammalian cells. So all tissues, all cells in the cytosol. What is a substrate? The substrate is glucose. So glucose yung substrate. So kailan ba maraming glucose? Kapag bagong kain ba tayo or pag fasting? Siyempre pag bagong kain. That's what we call the fed state or the absorptive state. So in the fed state or the absorptive state, that's the first 4 hours after a meal. Anong hormone yung mataas when you are in the fed state? It's insulin. And what's the second messenger of insulin? Tyrosine kinase. So in the fed state, the first 4 hours after a meal, that's what you call the absorptive state, insulin is high. And insulin is high, glucose is high, you will activate glycolysis. So, yun lang yung pinaka dapat nyong matandaan. In contrast, kapag fasting, wala tayong glucose noon. That's 4 hours after a meal. 4 hours after a meal. Dahil wala ng glucose, which is the substrate for glycolysis, inhibited naman siya. What are the end products? Glycolysis can produce either pyruvate or lactate. We will discuss later which is which. And then the rate-limiting step is the conversion of fructose 6-phosphate to fructose 1,6-bisphosphate, catalyzed by the enzyme phosphofructokinase 1 or PFK1. So this is glycolysis. So you will see here, glycolysis begins with glucose. And the product is either pyruvate or lactate. So we divide glycolysis into 2. The first 3 steps, that's the energy investment phase. In this phase, we consume 2 ATPs. And then the last 7 will be the energy generation phase. Ito naman, we produce 4 ATPs and 2 NADH. Para lang yung negosyo. Kung gusto nating kumita ng pera or ATP, kailangan muna nating mag-invest. Okay, kailangan muna nating mag-invest. As you can see here in this diagram, glycolysis consists of multiple steps. Question: lahat ba ng steps na yan, lahat ba ng enzymes negative yung delta G? No, hindi kailangan. Kasi sabi natin kanina, anong kailangan lang mangyari? Dapat lang when we add the different delta Gs, the overall sum should be negative for this pathway to proceed. So in the energy investment phase, what happens? Kailangan muna natin gumamit. Gagastos muna tayo ng 2 ATP. So you see here in step number 1 and in step number 3, okay, we use 1 ATP for each step. Kaya 2 ATPs in total yung ini-invest natin. And then afterwards, pagkatapos nating mag-invest, tsaka na tayo kikita. Tsaka na tayo kikita. So you will see here, there are 3 steps. 2 steps will yield 2 ATPs each. And then 1 other step will produce NADH. So look, so the 2 steps, the 2 enzymes that produce ATP, phosphoglycerate kinase and pyruvate kinase. While the step that produces NADH, glyceraldehyde phosphate dehydrogenase. Now, I want you to focus on something. So take note, when you see here, okay? So if you look at the pathway, tataka kayo, bakit ganun? Bakit pagdating dito, lahat sila times 2? Okay? So you will see here, okay? So you will see everything here is times 2. Okay? Bakit sila lahat times 2? Ganito kasi yan. Supposedly, pagdating sa step na to, Okay? Si fructose 1,6-bisphosphatase, fructose 1,6-bisphosphate, isi-split siya into two. Okay? One will be DHAP and the other one is glyceraldehyde 3-phosphate. Pero si DHAP, no, this one, naka-interconvert siya sa glyceraldehyde 3-phosphate. No? So, instead na isulat ulit siya lahat dito, no? So, instead na yung isa DHAP, isa glyceraldehyde 3-phosphate, lahat na lang times 2. So, one on the left and one on the right. Okay? One on the left, one on the right. Kaya lahat yan times 2 from that point. Yun lang yung tatandaan nyo dyan. Kasi yung fructose 1,6-bisphosphate, this one, sinisplit natin. At pag in-split siya, interconvertible yung products niya. Yung DHAP and glyceraldehyde 3-phosphate. Tamad kasi yung nag-drawing. No? Instead na nag-drawing siya sa kaliwa at sa kanan, everything is just times 2. Okay? So, do not forget that. Yun yung reason kaya lahat dyan times 2. And also, as I said earlier, you don't need to memorize everything. You just need to know the important steps. And for glycolysis, there are 3 important steps. Step numbers 1, 3, and 10. Step number 1 is the phosphorylation of glucose. Step number 2 is the phosphorylation of fructose 6-phosphate. And step number 10 is the formation of pyruvate. Now remember this, when we say kinase, sa biochemistry, pag sinabi natin kinase, it adds a phosphate. So, pag magdadagdag siya ng phosphate, kinase yun. Pag magtatanggal ng phosphate, phosphatase naman. So, you see here, nasan ba yung 3 important steps? The 3 important steps are step number 1. In step 1, glucose is converted to glucose 6-phosphate. Anong ibig sabihin nun? No? Glucose to glucose 6-phosphate. So, si glucose, ibig sabihin ba nun, kakabitan natin ng 6 na phosphate? No. So when we say glucose to glucose 6-phosphate, we add just 1 phosphate molecule at position number 6. No? So kaya siya tinawag na glucose 6-phosphate. It has 1 phosphate molecule at position number 6. So that step is catalyzed by hexokinase or glucokinase. So you will see there, no, glucose is converted to glucose 6-phosphate. Afterwards, no, glucose 6-phosphate is converted to fructose 6-phosphate using an isomerase. Bakit naman siya tinawag na isomerase? Kasi nga, it converts glucose to its isomer fructose. So you will see here, right? That's the next step. The next important step is phosphofructokinase 1. So from fructose 6-phosphate, dadagdagan pa natin ng isa pang phosphate. Kaya, ang mangyayari, fructose 1,6-bisphosphate na siya. So, kaya siya tinawag na phosphofructokinase. It's a kinase. It adds phosphate to phosphofructose. So, yung phosphofructose, which is fructose 6-phosphate, kakabitan natin ng isa pang phosphate. Ang kalalabasan, 1,6-bisphosphate na. And then sabi natin anong gagawin? Si aldolase, isi-split niya si fructose 1,6-bisphosphate into DHAP and glyceraldehyde 3-phosphate. Now, si DHAP, nako-convert din siya sa glyceraldehyde 3-phosphate. So, dito, instead na magdodrawing tayo ng isa pa, no? Times 2 na lang lahat kasi nga tamad yung illustrator. It's a series of steps such that glyceraldehyde 3-phosphate and DHAP are eventually converted to phosphoenol pyruvate. Okay, until we reach the final step, no? And the final step is the conversion of phosphoenol pyruvate to pyruvate using pyruvate kinase. So remember these 3 steps, no? Step number 1, step number 3, and step number 10. Bakit kaya sila special? No? Bakit kaya sila special? So when you look at the diagram, look at hexokinase, glucokinase, phosphofructokinase, and pyruvate kinase step. Ano yung kakaiba sa kanila dun sa step nila compared with the rest of the pathway? So when you look at the arrows, anong napansin nyo? Dun sa important steps, you only have single arrows. Okay? You only have single arrows. Compare that with the rest of the pathway. The rest of the pathway contains double arrows. Pero yung tatlong to, single arrow sila. What does that mean? These pathways are irreversible. They are not reversible. Okay? So, kaya sila special. Irreversible sila. And again, what's the rate-limiting step? The rate-limiting step is phosphofructokinase 1, which converts fructose 6-phosphate to fructose 1,6-bisphosphate. So let's discuss the details. So first step, glucose to glucose 6-phosphate. This can be catalyzed by two enzymes. It can either be via hexokinase or glucokinase. Kailangan kabisado nyo kailan hexokinase, kailan glucokinase. So hexokinase is present in most tissues, whereas glucokinase is only found in the liver and in the pancreas. So you will see here, right? You will see here, magkaiba sila ng property. Magkaiba sila ng property. Now, you will have to go back, no? You will have to go back to the properties of your enzymes. So, you will see here, no? You will see here, your hexokinase has a low Km, okay, which means it's got a higher affinity for glucose. In contrast, glucokinase has a high Km but a lower affinity for glucose. Ano naman ibig sabihin nun? So kung babalala nyo ng enzyme kinetics, pag sinabi natin na Km, no? Low Km, high affinity. Pag high Km, low affinity. Ano ibig sabihin? Kunyari ako si glucose. Kanino ako lalapit? Kay hexokinase or kay glucokinase? Dalawa sila nasa harap ko. Lalapit ako kay hexokinase. Kasi si hexokinase, mas mababa yung Km niya compared to glucokinase. So, low Km, higher affinity. So, tandaan nyo yan, ha? Low Km, dun mas lalapit si substrate compared to the other enzyme. But the other difference is, glucokinase has a high Vmax. Meaning to say, mas mabilis siya mag-convert ng glucose to glucose 6-phosphate. Anong importance nun? So, when you look at this diagram, so again, look, what do? They convert glucose to glucose 6-phosphate. So, you see here, so glucose, we add the phosphate at position number 6. So, saan natin kinuha yung phosphate? From ATP. Kaya dito tayo mag-i-invest ng phosphate. So, using hexokinase or glucokinase. And this is what you call the Michaelis-Menten plot. So, kung naaalala niyo yung Michaelis-Menten plot, no? So, kung naaalala niyo yung Michaelis-Menten plot, okay? So, you see here, okay? Yung color red, okay? Yung color red, yun yung plot natin. Sorry, yung color red, this is the plot for glucokinase. Yung color blue, yun yung plot natin for hexokinase. So, compare natin sila, no? So, you will see, okay? Yung Vmax, sino mas mabilis magtrabaho? Mas mabilis magtrabaho si glucokinase. So, that's why here, look, yung velocity ng enzyme, no? Mas mabilis siya mag-convert to glucose 6-phosphate. So, enzyme activity, mas mabilis. In contrast, si hexokinase, no? Si hexokinase, mas mabagal. Now, what about Km? What is the definition of Km? Km is the amount of substrate present so that the velocity is half the maximum. So when you look at this diagram, so creating Vmax ni glucokinase, yung half ng velocity niya, so this is the half of the maximum velocity, and the substrate concentration at that point is this. So this is the Km of glucokinase. Si hexokinase, ito yung Vmax, we get half. What's the Km? Km is this. Anong ibig sabihin nito? No? Ibig sabihin niyan, kung mas mababa yung Km ni hexokinase, mas konting glucose yung kailangan natin para yung velocity niya 50% agad of maximum. Okay. So ulit, yung Km ni hexokinase, this one, no? Mas maliit. Ibig sabihin, mas konting glucose yung kailangan natin, no? Mas konting glucose yung kailangan natin bago siya magtrabaho. Kaya nga siya low Km, high affinity. Kung merong glucose, una siya agad pupunta kay hexokinase, no? Kasi hexokinase, kahit konti lang yung glucose, mag-work agad siya. In contrast, si glucokinase, kailangan mo ng medyo mas madaming glucose bago siya magtrabaho agad. So, bakit ganun? Bakit sila magkaiba? Kasi magkaiba sila ng function, no? So, si hexokinase, anong purpose niya? Si hexokinase, it is found in most tissues. Ang purpose niya is to make sure that the cell is able to meet its needs. So, kunyari, kung muscle cell siya, kailangan meron siya laging glucose, glucose 6-phosphate para ma-meet yung energy needs. Para meron siyang pang-glycolysis. Okay? In contrast, no? Si glucokinase naman, bakit niya phosphorylate si glucose? Phosphorylate ni glucokinase si glucose for storage. Para meron tayong gagamitin for future use. No? Para meron tayong gagamitin for future use. Kailan yun bang gagawin? Kailan tayo mag-store for future use? Pag marami lang glucose. No? Pag marami lang glucose. So, paano yun mas madaling matandaan? Okay? So, for example, ikaw, duty ka sa ER mamayang gabi. No? So, kung duty ka sa ER mamayang gabi, unfortunately, toxic, Friday night, sweldo, hindi umuulan, no? So, maraming trauma, no? Tunod-sunod yung mga pasyente. Kaso, ang baon mo lang ay Skyflakes, no? Ang baon mo lang ay Skyflakes. So, anong gagawin mo dun sa Skyflakes na yun? Pag kinain mo yung Skyflakes, saan mo gagamitin yung glucose na sa Skyflakes na yun? Gagamitin mo ba siya para meron kang energy tonight? Or gagamitin mo ba siya for storage for future use? Siyempre, gagamitin mo siya for your energy tonight, di ba? So, dahil kumain ka lang ng Skyflakes, gaano kataas yung blood sugar natin? Konti lang. Hindi ganun kataas. No? Hindi ganun kataas. Konti lang. Okay? At dahil konti lang, if you look at this graph, konti lang yung itataas ng glucose natin. Dahil konti lang yung tinaas ng glucose natin, saan pupunta si glucose? Kay hexokinase or kay glucokinase? Kay hexokinase. No? So remember, no? Kahit konti lang yung glucose, pwede na siyang ma-phosphorylate. At ang mas mag-phosphorylate sa kanila, si hexokinase, higher affinity. Kay hexokinase muna. Kasi nga, ang purpose ni hexokinase, i-phosphorylate niya si glucose to meet the cell's need. Okay? To meet the cell's need. Tatandaan na, so kapag Skyflakes lang ang baon mo, no, hexokinase lang magpo-phosphorylase niyan. Kasi kailangan na siya for metabolism that night. In contrast, halimbawa, duty ka ulit mamaya, pero birthday ng barkada mo. So, ano nangyari? Kumain kayo sa buffet. No? Kumain muna kayo sa buffet. So, dahil kumain kayo sa buffet, madaming kang glucose. So, pag tumaas ngayon yung glucose mo, no? Tataas ang glucose. Look at the graph. No? Tataas ngayon yung glucose. Maaabot na natin yung KM ni glucokinase. So, yung kinain mo sa buffet, gagamitin mo ba yun para lang sa duty mo tonight or magsa-store ka for future use? Some of it, you will store for future use. So that's why pag kumain tayo ng madami, tataas yung glucose concentration natin enough para maabot yung KM ni glucokinase. At doon na magtatrabaho si glucokinase. Ipo-phosphorylate niya si glucose where? In the liver and in the pancreas. And in the liver, that phosphorylated glucose will be used for storage for future use. Bakit kailangan mabilis siya magtrabaho? Bakit kailangan mataas yung Vmax? No? Kasi syempre, pag kumain tayo sa buffet, hindi naman permanently mataas yung glucose. Mataas lang yan for the next 2 to 4 hours. Kaya kailangan mabilis magtrabaho si glucokinase para mas marami tayong ma-phosphorylate. Okay? So, tatandaan, no? So, anong difference ng dalawa? So, hexokinase, low KM, high affinity. It's found in most tissues. Ang purpose niya, i-phosphorylate niya si glucose para meron tayong energy to meet the cell's need. In contrast, si glucokinase naman, no? So, si glucokinase, ano naman ang purpose niya sa buhay? Ang purpose naman sa buhay ni glucokinase, in the liver, in the pancreas, it will phosphorylate glucose to be stored for future use. So, for synthesis of glycogen and lipids. Si glucokinase, high KM, low affinity, but higher Vmax. So, paano natin yun matatandaan, no? So, tatandaan nyo, okay, H, okay, and G. Okay. So, para matandaan natin yan, remember, no? So, yung letter G, para yung kamay ng waiter, no? Para yung kamay ng waiter, meron siyang hawak na chicken. Okay? So, may hawak siya na chicken, letter G. So, kumain ka sa buffet. So, pag kumain ka sa buffet, sino ma-activate natin? Si glucokinase. Storage for future use. Okay? Pero, ano naman to? Okay. Yan ay Skyflakes. So, pag Skyflakes lang ang baon mo sa duty, sino lang ma-activate natin? Si hexokinase. Now, what about step number 3? The next step is the rate-limiting step of glycolysis. So again, rate-limiting step, ito yung pinaka-important na step. This converts fructose 6-phosphate to fructose 1,6-bisphosphate. So, from 1-phosphate molecule, gagawin nating dalawa. Yan siya, phosphofructokinase. It's a kinase. It will add phosphate to phosphofructose. So, you will see here, that's the reaction in the screen. Now, titingnan nyo to, no? Fructose 6-phosphate to fructose 1,6-bisphosphate. Pero look, what else is here? No? You see is fructose 2,6-bisphosphate. No? Para siyang 1,6-bisphosphate, pero anong kaibahan? Yung fructose, yung phosphate niya, nasa position 2 and position 6. Anong ginagawa niya? Ang ginagawa niya, ina-activate niya yung reaction. Pinapabilis niya yung reaction. Diba? Pinapabilis niya yung reaction. So, meron kasi tayong dalawang phosphofructokinase, no? So, meron tayong dalawang phosphofructokinase. Phosphofructokinase 1 and phosphofructokinase 2. So, PFK1 and PFK2. Ano pinagkaiba nila? Yung rate-limiting step, si PFK1. No? So, PFK1 produces fructose 1,6-bisphosphate. Yun yung rate-limiting step, no, ng glycolysis. Yun yung nasa pathway. Anong silbi ni PFK2? Si PFK2, tinutulungan niya si PFK1. Paano? So, PFK2 produces fructose 2,6-bisphosphate, which is the most important activator of PFK1. Okay, so tatandaan nyo ha. So, si PFK2, tinutulungan niya si PFK1. Paano? By producing fructose 2,6-bisphosphate, which is the most important activator of PFK1. Bakit natin gagawin yon? Kasi gusto natin, no, pag magre-release ng energy, mabilis. Rapid release of energy. Ito yung feed forward. Pag magme-metabolize tayo ng glucose, pag magme-metabolize tayo ng glucose, mabilis tayo mag-metabolize para lumabas, ma-release agad natin yung energy. Makakuha agad tayo ng ATP. That's feed forward regulation. So, kumbaga, pinapabilis niya yung sarili niya. So, if they ask you, tandaan, what's the most important activator of PFK1? The answer is not PFK2. The answer is fructose 2,6-bisphosphate, which is produced by PFK2, phosphofructokinase 2. And if they ask you what's the rate-limiting step of glycolysis, it's phosphofructokinase 1. Finally, the last important step is step number 10, which converts phosphoenol pyruvate to pyruvate using the enzyme pyruvate kinase. Now, this is not the first time you heard phosphoenol pyruvate, diba? Kailan nyo narinig si phosphoenol pyruvate? Yan yung kumpare yung maraming pera, pards! Long time no see, pautang naman. O, diba? Siya yung mayaman. No? You see? Yung mayaman, yung yaman niyang phosphate, uutangin natin para makaproduce ng ATP. So, phosphoenol pyruvate is converted to pyruvate, and this step will produce 1 ATP per molecule of phosphoenol pyruvate. So, ilan total mapoproduce? Sa step na yan, dalawa ha. So, bakit dalawa? One on the left and one on the right. What kind of ATP production is this? What kind of ATP production is this? Is this substrate level phosphorylation or oxidative phosphorylation? This is substrate level phosphorylation. Diba? Nangutang lang tayo ng phosphate. Hindi naman tayo dumaan sa ETC tsaka oxidative phosphorylation. Okay, sa ATP synthase. So actually, there are two steps. No? There are two steps where substrate level phosphorylation occurs. The first is the phosphoglycerate kinase step. And the second one is the pyruvate kinase step. So if you look at the diagram again, so you see here, these are the two steps that produce ATP. 2 ATPs each. One on the left and one on the right. At ang donor ng phosphate, si 1,3-bis phosphoglycerate, tsaka si phosphoenol pyruvate. Yan yung mga kaibigan nating mas mayaman. So if you remember, no, sila yun. So in the energy generation phase, we produce how many ATPs? 4. So 2 steps that produce 4 ATPs each. by borrowing phosphate from these high-energy compounds. So, the phosphate is transferred from phosphoenol pyruvate and 1,3-bis phosphoglycerate to ADP, forming ATP. Aside from ATP, we also produce NADH. Oh, anong isipin nyo pag nakakita kayo ng NADH? Ang maisip nyo ay tseke, no? So nakaka-produce din tayo ng tseke sa glycolysis at important 'yon, no? In this step, your glyceride 3-phosphate dehydrogenase step, we are able to produce 2 molecules of NADH. Bakit important 'yan? Bakit important yung tandaan? Kasi yung NADH na yan, anong pwede natin gawin? Pwede natin yung encash para maging ATP. O, review. Saan nangyayari yung glycolysis? What part of the cell? It happens in the cytosol, no? Pero nasan yung bangko? Nasa mitochondria, in the electron transport chain. Paano ngayon natin dadalhin yung tseke from the cytosol to mitochondria? That is what we will discuss next. So again, quick rundown. Ilang ATP yung ininvest for glycolysis? Dalawa. Okay? Ilang ATP naman yung kikitain? 4. 2 steps of substrate level phosphorylation. How about NADH? 2. No, your glyceride phosphate dehydrogenase step produces 2 NADH molecules, one on the left, one on the right. Dito tayo magkakaroon ng sinasabing 2 types of glycolysis. So you see here, when you convert glucose to pyruvate, that is what we call aerobic glycolysis. So you will see in cells with mitochondria and cells with adequate oxygen supply, the end product of glycolysis is pyruvate. So glucose to pyruvate. So the last step is the conversion of phosphoenol pyruvate to pyruvate using the enzyme pyruvate kinase. However, if you do not have oxygen or if you do not have mitochondria, the end product of glycolysis is lactate. So yung pyruvate, i-convert pa natin yun to lactate using lactate dehydrogenase. Kailan nangyayari yun? When you don't have oxygen or you don't have mitochondria. So, dito muna tayo sa aerobic glycolysis. Sabi natin, aerobic glycolysis, you need oxygen, you need mitochondria. Ano nangyayari dito? Yung check-in natin yung NADH. Diba? Sabi natin, uy, may NADH tayo na na-produce sa glycolysis. Tuwang-tuwa tayo, yay, may check-in ako, NADH. Kailangan natin yung dalhin sa bangko. No? Ang bangko, nasa mitochondria. Pero nandun tayo sa cytosol. Bakit yung problem? If you remember, yung mitochondria may dalawang membrane. May inner membrane, tsaka outer membrane. Si outer membrane, palulusutin tayo nun. Palulusutin tayo ng outer membrane. Pero si inner membrane, impermeable. So you can see here, ito yung mga check-in natin, NADH, na na-produce sa glycolysis. Kailangan natin pumasok sa loob kasi nandun yung bangko. However, ayaw tayong papasukin nito. Sino to? Si inner membrane. So, anong kailangan natin gawin? Para makapasok tayo sa loob, no? Para makapasok tayo sa loob, anong kailangan? Kailangan nating sumakay sa taxi, no? So, you will see from here, sasakay tayo ng taxi. Okay, yan. Okay? So, kailangan nating sumakay ng taxi para makapasok tayo sa mitochondria. Okay? So, meron tayong dalawang choices. So, you see here, dalawang choices yung pwede nating sakyan. We can either ride the glycerophosphate shuttle, no? Or the malate aspartate shuttle. Okay, so either glycerophosphate shuttle or malate aspartate shuttle. So, anong pinagkaiba nung dalawa? So, actually, wag pala taxi, bus, no? Mas kasi shuttle, no? Para masanay tayo, shuttle bus, no? So, dalawang bus. So, pwede tayong tumakay sa glycerol phosphate o pwede tayong sumakay sa malate aspartate shuttle. So, you will see here, pag tumakay tayo ng bus, so, wag taxi, sakay tayo ng bus. Yan. Okay? So, pwede tayong sumakay either dito, kay malate aspartate shuttle, or glycerophosphate shuttle. So si malate aspartate shuttle, you will see sasakay tayo as NADH, no? So sasakay tayo as NADH. Tapos dahil nakasakay na tayo sa bus, pwede na tayong pumasok sa loob. Pagdating sa loob, anong sasabihin nila? Oh, mitochondria na po, mitochondria na. Lalabas na ulit si NADH. Pupunta na siya ngayon sa complex 1. Tama. Okay, so yung NADH, synthesitosol, sasakay ng bus, papasok sa loob, papunta ngayon sa mitochondria, papasok sa complex 1. Very good. Ilang ATPs yung makukuha natin? 3. Bakit 3? So, papasok tayo sa complex 1, pasa-pasa sila ng electron. Sino yung uutot? Complex 1, 3, and 4. Ilang protons ang ma-generate? 2.5. 1, 1, and 0.5. Ano mangyayari sa protons? Gusto ba nila sa intermembranous space? No. Kailangan nilang bumalik sa matrix. At para makabalik sa matrix, kanino sila dadaan? Okay, complex 5, also known as ATP synthase. So pag lusot nila sa ATP synthase, ting! You've just produced ATP. Makakakuha tayo ngayon ng 2.5 ATPs for every NADH. At ilang NADH ba yung sasakay sa bus? 2. Kaya ilang ATP siya makukuha natin dyan kung sumakay tayo ng malate aspartate shuttle? 5. No? So 2.5 times 2. Paano kung puno na yung malate aspartate shuttle? Kung puno na yung malate aspartate shuttle, no? Sasakay tayo kay glycerophosphate shuttle. So you will see, pwede din tayo sumakay kay glycerophosphate shuttle. Anong problem kay glycerophosphate shuttle? Unfortunately, itong si glycerophosphate shuttle, pag doon tayo sumakay, maho-hold up tayo. No? So you will see, sasakay kang NADH, pero pagdating sa mitochondria, FADH ka na lang. No? Pagbaba mo, na-hold up tayo. No? Bakit tayo lugi nun? Kasi pag sumakay kang NADH pero bumaba ka ng FADH, lugi yun kasi si FADH kaninong teller pupunta? Kay complex 2. At kapag kay complex 2 tayo pumasok, ilang ATPs lang yung mapoproduce natin? 1.5 for every FADH molecule. Kaya kanina, nakuha natin 5 ATPs. Ngayon, ilan na lang makukuha natin? Just 3. No? Still, hindi mo tatanggihan yun. No? Still, hindi mo yun tatanggihan kasi ATPs pa din yun. So, once again, yung NADH na na-produce natin ng glycolysis, kapag aerobic conditions, anong gusto natin gawin? Gusto natin silang dalhin sa mitochondria para maging ATP. Bakit tayo may problem? Kasi si inner membrane hindi tayo basta-basta papapasukin. So anong gagawin natin? Sasakay tayo ng bus. We can either take the malate aspartate or the glycerophosphate shuttle. So in the heart and most tissues, we will take the malate aspartate shuttle. In brain and white muscle, we will take the glycerophosphate shuttle. However, you must remember, ano nangyayari kay glycerophosphate shuttle? Tayo ay naho-hold up. Sasakay kang NADH, bababa kang FADH na lang. Kaya sa malate aspartate shuttle, ilang ATP sa makukuha natin in total? 5. Kasi 2 molecules of NADH times 2.5. Pero pag sumakay tayo ng glycerophosphate shuttle, ilang ATPs na lang makukuha natin? 3. Kasi 1.5 times 2. Anong mnemonic? Malate aspartate shuttle, mas safe. No? Malate aspartate shuttle, mas safe. Kasi sa glycerophosphate shuttle, na-hold up tayo. Okay, so mamaya, ito total natin ilang ATPs na lahat-lahat therefore. Okay? So kung hindi tayo aerobic glycolysis, anaerobic glycolysis ang mangyayari. So in this scenario, walang oxygen or walang mitochondria. So yung NADH na na-produce natin sa glycolysis, kung walang mitochondria, mapapa-encash mo ba? No, di ba? Yung NADH, walang silbi yan. Hindi natin mapapa-encash kasi walang mitochondria eh. No? Walang mitochondria. Kung may mitochondria pero wala namang oxygen, may mitochondria ka nga, wala namang oxygen, tatanggapin ba siya ng ETC? Hindi rin. Why? Kasi ano bang role ni oxygen? Siya yung final electron acceptor. Remember? Siya Yung final electron acceptor. So therefore, yung NADH, no? Yung NADH kapag, no? Okay, so let's see. So si NADH, you see this? Kapag walang oxygen or walang mitochondria, hindi siya nadadala sa bangko. Instead, we will use it to convert pyruvate to lactate, as you can see on the screen. So, dahil nga wala naman tayong pagdadalhan, yung pyruvate, i-convert na lang natin siya to lactate. Doon natin nagagamit yung NADH. That's why in anaerobic glycolysis, what's the end product? It's not pyruvate but lactate. So, 2 molecules of pyruvate will be converted to 2 molecules of lactate using the enzyme lactate dehydrogenase. So, kailan nangyayari to? So, nangyayari to pag walang oxygen or walang mitochondria. So, for example, red blood cells. Again, what kind of glycolysis will red blood cells undergo? Anaerobic glycolysis because mature red cells do not have mitochondria. The end product of glycolysis will always be lactate. Ordinarily, your muscle, if it's well oxygenated, it will undergo aerobic glycolysis. Pero kunyari nag-exercise ka ng exercise, tumakbo ka ng marathon, hindi ka naman ready. Maubusan ka ng oxygen. Your muscle cells will shift from aerobic metabolism to anaerobic metabolism. And what will happen? You will generate lactic acid and that can cause cramps. Brain. Your brain, initially, well oxygenated. Pag nag-stroke, mag-shift siya to anaerobic metabolism. And therefore, the product of glycolysis will no longer be pyruvate. Instead, the product will be lactate. So let's compute how many ATPs are produced. On the left column, you will see aerobic glycolysis. On the right, you will see anaerobic. Kaya pag aerobic glycolysis, ilang ATPs yung makukuha natin, no? So you will see, kapag aerobic glycolysis, okay, 4 ATPs will be produced from substrate level phosphorylation. Bakit 4? Kasi 'di ba, dalawang steps that will produce 2 ATPs each. Pero tatandaan nyo, nag-invest tayo ng dalawa sa step number 2 at step number 3. Kaya kung glycolysis lang yung pag-uusapan, no? Kung glycolysis lang pag-uusapan, dalawang ATPs net, no? So 4 minus 2. However, you must remember that we also can get ATPs from NADH. So, 3 or 5. So, depende kung anong shuttle. Kapag malate aspartate, we get 5. Kapag glycerophosphate, you get 3. Ano ibig sabihin nun? In total, in aerobic glycolysis, you can either get 5 or 7 ATPs. So, pag pinag-add natin yung lahat. Okay? Depending on the shuttle. In contrast, kapag anaerobic glycolysis, parehas yung dalawa, no? You earn 4, you invest 2. Pero, no? You will see you will not be able to generate any ATP from NADH. Wala tayong makukuhang ATP from NADH. Okay? And that's the reason why if you undergo anaerobic glycolysis, you only get a net of 2 ATPs. Kasi 4 minus 2. So, kaya nga sabi ko, di ba? In aerobic glycolysis, no, mas madami tayong nakukuhang energy. In physiology, you've probably heard about 2,3-BPG or 2,3-bisphosphoglycerate. So, ito yung sa carbon dioxide, dun sa oxygen hemoglobin dissociation curve. No? So, yung sa oxygen hemoglobin dissociation curve, shift to the left, shift to the right. Other name nito, 2,3-DPG. Yun yung old name niya. Anong effect ng 2,3-BPG? If you remember, 2,3-BPG will cause, no, your oxygen hemoglobin dissociation curve to shift to the right. No, ibig sabihin, pag nandyan si 2,3-BPG, mas readily nating i-release. Mas readily nating i-release yung oxygen to the peripheral tissues. So that 2,3-BPG actually comes from glycolysis. So this is the Rappaport-Lubbering pathway. So you will see here, okay? So this is how you get 2,3-BPG. So in glycolysis, the 1,3-BPG is converted to 2,3-BPG using phosphoglycerate mutase, no, or biphosphoglycerate mutase. Yan yung pangalan niya, no? So that's the Rappaport-Luberin pathway. Just something to remember. Okay? So pyruvate, it's the end product of aerobic glycolysis. Ano ngayon ang mangyayari kay pyruvate? So si pyruvate, no, parang kayo yan. No? Marami siyang pwedeng puntahan. So after medical school, anong pwede mong gawin? Pwede kang mag-residency, pwede kang mag-general medicine, pwede kang mag-doctor to the barrio, pwedeng hindi ka magpaka-doctor at all. No, pwedeng mag-business ka na lang. Or pwedeng mag-influencer. Or pwedeng decide ka magpaka-houseband or housewife. Di ba? So, and then you do other things like research, teaching. So, marami tayong pwedeng puntahan. So, parang ganun din si pyruvate. So, si pyruvate, after glycolysis, marami siyang choices. And you need to be familiar what happens to pyruvate. So, kakabisaduhin niyo tong crossroads na to. So, pyruvate can become lactate. Anong enzyme? Lactate dehydrogenase. Kailan nangyayari to? This happens when we don't have oxygen or mitochondria. So, this happens when we don't have oxygen or mitochondria. We go up. Pyruvate aakyat. Pero, kung meron naman tayong oxygen at meron namang mitochondria, hindi tayo pupunta sa anaerobic glycolysis. Saan tayo pupunta? Si pyruvate, bababa siya. Okay, so kapag may oxygen, may mitochondria, pyruvate will go down. Using pyruvate dehydrogenase, we will convert pyruvate to acetyl-CoA. Bakit natin gagawin to? So that acetyl-CoA can enter the Krebs cycle. And there, we will produce even more ATPs. Again, walang oxygen, walang mitochondria, akyat tayo sa lactate. Pero pag may oxygen, may mitochondria, baba tayo sa acetyl-CoA so that we can enter the Krebs cycle. Now, pyruvate, if you recall, has 3 carbons. Kaya nga 6 carbons ng glucose, magiging 2 molecules of pyruvate. Yung 3 carbons na yun, pwede nating dagdagan. So, it's a carboxylation reaction, no? So, yung carboxylation reaction, pyruvate carboxylase. So, 3 carbons magiging oxaloacetate, which has 4 carbons. This happens in gluconeogenesis. So, we will discuss this next. In contrast, pwede rin natin siyang bawasan, pyruvate decarboxylase. So, from 3 carbons, magiging 2 carbons na lang, ethanol. Sino naman to? Ito naman yung fermentation, no? So, can you do this? No. Unless you are yeast, no? So, yeast lang yung nakakagawa nito, no? So, yeast yung nakakagawa nito. So, they convert pyruvate to ethanol, and this is how you produce liquor. This is how you produce alcoholic beverages. So, we ferment pyruvate into ethanol. So, wag kakalimutan, pyruvate going up, lactate. Pyruvate going down, acetyl-CoA. Going to the right, oxaloacetate. Going to the left, ethanol. Remember the enzymes, remember the pathway. So, kakadiscuss lang natin yung going up. Yung going up, nangyayari pag walang oxygen, walang mitochondria. Kailan naman tayo bababa? O, di ba sa business, ano sabi natin? Sa business, kapag kumita ka na ng pera, ano nang gusto mong gawin? No? So, kumita ka na ng pera, ano nang gusto mong gawin? Hindi gumastos, no? Hindi ka negosyante, no? Kapag gusto mo gumastos. Pag pumasok sa isip mo, gumastos, hindi ka negosyante. Sa business, kapag kumita ka na ng pera, gusto mo kumita pa ng mas malaki. O, di ba? Sa glycolysis, sabi natin, pag anaerobic glycolysis, how many ATPs in total? 2. Pag aerobic glycolysis, how many ATPs in total? 5 or 7, depending on the shuttle. So, pag kumita na tayo ng 5 or 7 ATPs, hindi pa tayo kontento doon. Gusto natin kumita pa tayo ng mas malaki. So, yung pyruvate, i-convert natin siya to acetyl-CoA. Para pag na-convert natin siya sa acetyl-CoA, ano mangyayari? Pwede na tayong pumasok sa Krebs cycle. And that's where we will produce a lot more ATPs, as I will show you later. Okay? So, ito yung reaction na dapat yung tandaan. The conversion of pyruvate to acetyl-CoA is catalyzed by pyruvate dehydrogenase. So, you will see here, pyruvate to acetyl-CoA requires pyruvate dehydrogenase. And you will see also here, no, it produces NADH. So, So this reaction produces NADH and releases carbon dioxide. This is catalyzed sa pyruvate dehydrogenase complex enzyme. Now, si pyruvate dehydrogenase sa buong biochemistry, siya yung pinakamaarte sa lahat. No, siya yung pinakamaarte. Huwag niyong kakalimutan yan. At bakit siya maarte? It has 5 cofactors or coenzymes. And all of these enzymes have to be present. Pag kulang yan kahit isa, anong sasabihin ni pyruvate dehydrogenase? Ay, ayoko nga. Hindi siya mag-work. It will not work at all. Hindi niya i-convert si pyruvate to acetyl-CoA. Hindi tayo makakapag-Krebs cycle. Ano mangyayari sa pyruvate? Dahil hindi siya nagiging acetyl-CoA, hindi siya makababa. Saan siya pupunta? Sa taas. It will be converted to lactate. So what are the 5 cofactors? The 5 cofactors are thiamine, FAD, NAD, coenzyme A, and lipoic acid. Once again, thiamine, FAD, NAD, coenzyme A, and lipoic acid. To make it easier to remember, these are the first 4 B vitamins with lipoic acid. So thiamine is vitamin B1. FAD is derived from vitamin B2, riboflavin. NAD is derived from niacin, vitamin B3. And coenzyme A contains pantothenic acid, which is vitamin B5. So B1, B2, B3, B5, and lipoic acid. The other thing you need to remember is that lipoic acid can bind to arsenic. Pag si lipoic acid nag-bind kay arsenic, wala na siyang silbi. Magtatrabaho ba si pyruvate dehydrogenase? No. Sabi niya, ay, ayoko nga. Kasi nga, walang lipoic acid. No, kailangan present silang lahat. Kaya si pyruvate dehydrogenase, siya yung pinakamaarte sa lahat ng enzymes because it requires 5 cofactors. So let's go to clinical correlates. So this is the first table of its kind. No? So ito, marami kayong makikitang table na ganito sa handout where you have an enzyme deficiency, clinical presentation, and some clinical comments. So ano bang ibig sabihin nito? No? So you will see. So most biochemical disorders, if you remember, are because of enzyme deficiencies. No? So for example, if you have a substrate here, no, you have a substrate that's being converted to a product using an enzyme. So kung itong enzyme wala, ano mangyayari? If you do not have that enzyme, the level of the substrate will go up. Tataas siya. How about the product? The product will go down. Okay. Kaya tayo nagmamanifest with clinical symptoms. No? So, kaya tayo nagkakaroon ng clinical manifestations. So for example, sa phenylketonuria. So in phenylketonuria, you're supposed to convert phenylalanine to tyrosine. No? So dapat i-convert natin yung phenylalanine to tyrosine using phenylalanine hydroxylase. So kung walang phenylalanine hydroxylase, what will happen to tyrosine levels? Tyrosine levels will go down. Yung phenylalanine levels naman tataas and it's converted to phenylketones which will become toxic to the body. So pag may enzyme deficiency, anong ginagawa natin? Pag may enzyme deficiency, either papalitan natin yung enzyme or isusupplement natin yung product. So in this case, tyrosine. Bibigay natin ng tyrosine yung patient. And iiwasan natin yung phenylalanine. So bawal yung mga may phenylalanine. Okay, so lagi nyo lang tatandaan yan, no? Yung basic principle na yan in biochemistry. So for example here, if you see a table like this, I don't want you to memorize this table row by row, cell by cell. What is more important for me is that you understand what's happening. Okay, so for example, puntahan natin si pyruvate dehydrogenase. So what will happen if you do not have pyruvate dehydrogenase? So recall, para saan ba si pyruvate dehydrogenase? Pyruvate dehydrogenase converts pyruvate to acetyl-CoA. So si pyruvate, i-convert to acetyl-CoA para makapasok sa Krebs cycle. If you do not have pyruvate dehydrogenase, what will happen? Si pyruvate hindi makakababa sa acetyl-CoA. Saan siya pupunta? Aakyat siya to lactate. No, it cannot go down, so pyruvate will go up. Kaya pyruvate dehydrogenase deficiency manifests as congenital lactic acidosis. So yung clinical scenario nito, yung baby, bagong panganak, and then severe lactic acidosis, elevated lactate. So how do we treat it? So ang nangyayari, no? Yung mga patients na to, dahil wala silang acetyl-CoA is also needed for acetylcholine. So wala silang neurotransmitter. So you will have psychomotor retardation and death. So we treat them using a ketogenic diet. How about pyruvate kinase deficiency naman? Yung pyruvate kinase deficiency, para saan ba si pyruvate kinase? Remember, pyruvate kinase is the step number 10 of glycolysis. Kung walang step number 10, walang pyruvate kinase, ano mangyayari? Mababawasan tayo ng step for ATP production. So remember, pyruvate kinase generates 2 ATPs, left and right. So kung wala yon, pag nag-glycolysis ka, nag-invest ka ng dalawa na lang din ang kikitain mo. So ang net worth mo, ang net income mo ay 0. So 2 plus 2 minus 2 na lang ang mangyayari instead na 4 minus 2. And kanino yun very harmful? For your red blood cells. So yung red blood cells kasi nakadepend sila sa anaerobic glycolysis. Kung wala kang pyruvate kinase, hindi na sila kikita, no? Hindi na sila makakakuha ng 2 ATPs for every molecule of glucose. 0 na. And why is that bad? Kasi yung red blood cells, kailangan nila yung ATP to maintain their membrane potential. If you remember your sodium-potassium ATPase pump, kailangan natin ng ATP to maintain the membrane potential. And because the red blood cells are not able to maintain their membrane potential, they are prone to hemolysis. No? So, sobrang fragile ng membranes. Muscle phosphofructokinase deficiency, PFK is the rate-limiting step of glycolysis. So, kung wala tayong PFK1, impaired yung glycolysis natin. So, si muscle, no, kahit na marami kang ininom na glucose, energy drink, di naman niya magagamit yung glucose to release ATP, leading to low exercise capacity, especially kapag high carbohydrate diet. Anong treatment? We will provide fats or lipids as alternative fuel. So we now go to the Krebs cycle. So the Krebs cycle, also known as the citric acid cycle or the tricarboxylic acid cycle. It's the final common pathway for the aerobic oxidation of carbohydrates, lipids, and proteins. So if you remember earlier, I told you, when you digest carbohydrates, proteins, and fats, the pathways converge to the molecule acetyl-CoA, and then acetyl-CoA will enter Krebs cycle. And also, if you remember earlier, yung Krebs cycle, anong classification niya? Is it anabolic or catabolic? It's amphibolic. So whether you're fasting or well-fed, tuloy-tuloy lang yung Krebs cycle. Tuloy-tuloy lang siya. So what is it for? This is the major pathway for formation of ATP. Ibig sabihin, karamihan ng ATP natin dito nanggagaling. This is also the pathway that provides a substrate for gluconeogenesis, amino acid synthesis, and fatty acid synthesis. So when we synthesize fat, when we create glucose, when we create proteins, kailangan natin ng Krebs cycle. It occurs in all cells with mitochondria. So, kailangan natin ng mitochondria. Without mitochondria, we will not be able to undergo Krebs cycle. Specifically, it occurs in the mitochondrial matrix, except for the succinate dehydrogenase step, which is in the inner mitochondrial membrane. The substrate, we begin with acetyl-CoA. So, saan nanggagaling si acetyl-CoA during the well-fed state? Siyempre, glycolysis, yung palang na-discuss natin na pathway, di yun lang yung sagot, So, pyruvate is converted to acetyl-CoA using pyruvate dehydrogenase step, and that is the source of acetyl-CoA when we are well-fed. How about when we are fasting? During the fasting state, this acetyl-CoA will come from beta oxidation, or the breakdown of fatty acids. The products are carbon dioxide, GTP, NADH, and FADH, as I will show in the next few slides. The rate-limiting step is the conversion of isocitrate to alpha-ketoglutarate using isocitrate dehydrogenase. So, this is the Krebs cycle. I'm sure many of you are intimidated by this, but I hope to show you na actually madali naman siyang maintindihan. So, don't focus on memorizing the individual steps. You just need to understand what's happening. So, ang starting point natin si acetyl-CoA, and you will see it will combine with oxaloacetate. So, acetyl-CoA, remember, has 2 carbons. So, acetyl-CoA, 2 carbons. It will combine with oxaloacetate 4. So 2 plus 4 equals 6. So 2 plus 4 equals 6 citrate. No? So 2 plus 4 equals 6. Yung citrate, 6 carbons na yan. Right? Tapos, ano mangyayari? Si citrate, i-rearrange siya to isocitrate. From isocitrate, ano mangyayari? Magbabawas ng isang carbon. So 6 minus 1, 5 carbons na lang, alpha-ketoglutarate. And then from alpha-ketoglutarate, bawasan mo na naman ng isang carbon, succinyl-CoA na siya. And then succinyl-CoA converted to succinate, fumarate, malate, balik na naman tayo sa oxaloacetate. So essentially, ano nangyari? You will see si acetyl-CoA na 2 carbons, umikot tayo sa Krebs cycle para makaproduce ng 2 carbon dioxide. Okay? So, ano ba yung carbon dioxide na yan? Anong gagawin natin dyan? No? Anong gagawin natin dyan? Inhale, exhale. O yan, yung carbon dioxide na yan, galing yan sa Krebs cycle. So, yung ini-exhale natin na carbon dioxide, dito yun ang galing. So, pag kumain ka, yung acetyl-CoA, na galing sa kinain mo, no, papasok sa Krebs cycle, magpo-produce ng carbon dioxide. So, para lang tong si acetyl-CoA, para siyang dumadating sa airport. So, pagdating niya sa airport, susunduin siya ni oxaloacetate. Tara, ikot tayo. Iikot siya ni acetyl-CoA, ni oxaloacetate. Tapos magiging carbon dioxide siya. Kaya matatandaan nyo lagi, no, ilang carbon dioxide pinoproduce sa Krebs cycle? 2 lang. Bakit 2 lang? Kasi 2 carbon lang yung pumasok eh. Hindi 2 carbon lang din yung lalabas. Okay? Is that clear? Now, ano ngayon? Anong silbi? Bakit pa siya iikot? Kasi hindi lang carbon dioxide yung napoproduce. Every time iikot si acetyl-CoA sa Krebs cycle, you will also produce, look, 3 molecules of NADH and 1 molecule of FADH. Anong sabi ko sa inyo? Every time you see NADH and FADH, anong maisip natin? Cheque. Pwede natin yang gawing pera. So, anong kailangan natin gawin? Dadali natin sila sa electron transport chain sa bangko para maging ATP. So, yung NADH natin, bawat NADH molecule, mako-convert natin to how many ATPs? 2.5. Whereas yung FADH, mako-convert natin to how many ATPs? 1.5. No? 1.5 naman. So, can you imagine, every time iikot si acetyl-CoA sa Krebs cycle, pag may pinasok tayong pag may pinasok tayong acetyl-CoA sa Krebs cycle, kikita tayo ng maraming pera. Ang tanong, no, this is the question. Yung NADH at FADH na to, no, para mapa-encash natin sa bangko, kailangan ba nating sumakay ng shuttle kagaya kanina? Okay. The correct answer is no. Why not? Because remember, saan ba matatagpuan yung bangko? Sa inner mitochondrial membrane. Saan ba nangyayari yung Krebs cycle? Sa mitochondria din, in the mitochondrial matrix. So, ibig sabihin, no, tatawid ka na lang, bangko na. Wala na tayong kailangang tawirin, no? Wala na tayong kailangang i-cross na membrane. So, we're already in the mitochondria. Hindi kagaya kanina. Kasi kanina, sa glycolysis, nasa cytosol tayo. Kailangan natin pumasok ng mitochondria. Kaya tatanungin kita, anong shuttle? Pero this time, dahil nasa loob na tayo ng mitochondria, hindi na natin to kailangan ng shuttle. Maglalakad ka na lang. Bangko na. Okay? So now, again, no? So can you try to imagine, no, mamaya kukumpitin natin yung ATP na napoproduce dito. And then finally, we also produce, no, 1 molecule of GTP. Guanosine triphosphate is an ATP equivalent. Meaning to say, pwede mo yung ipaswap. 1 GTP equals 1 ATP. Kaya energy ulit siya. No? So paano nyo matatandaan? Saan ba tayo magpo-produce ng GTP? Sa Krebs cycle. Tatandaan nyo lang, anong lumalabas sa susu? Gatas, no? Susu, gatas. So, succinyl-CoA to succinate, that's where you produce GTP. So, how do you memorize this? So, I'm sure you've seen a lot. Marami kasing mnemonic dyan, no? So, maraming mnemonic na pwede kayong gamitin, no? So, maraming mnemonic na pwede kayong gamitin para yun muna yung ma-memorize nyo. Just memorize first the elements. So ito yung mnemonic namin, pre-med pa ako, no? On campus, I kiss sweet, simple, fair maiden, no? So on campus, I kiss sweet, simple, fair maiden. So dapat with consent, no? Dapat with consent. Okay, so I'm sure there are many other mnemonics that you can use. Use the one that adheres to your moral standards. Okay? So, next. So, after nun, tatandaan nyo, itong mga merong napoproduce na NADH tsaka FADH. Ang pangalan ng lahat ng enzyme na yan, dehydrogenase. So, for example, here, no? So, ito, ang pangalan ng enzyme na to, isocitrate dehydrogenase. Ito naman, alpha-ketoglutarate dehydrogenase. Ito naman, malate dehydrogenase. So, basta may NADH or FADH, dehydrogenase ang pangalan yan. And this one, succinate dehydrogenase. So, pag walang NADH, FADH, hindi yun dehydrogenase, ha? So, yung mga dehydrogenase, yun lang yung nagpo-produce ng NADH and FADH. Kasi yun yung mga redox reactions. So, paano nyo matatandaan saan lalabas si FADH? Madali lang din, no? Madali lang din. Tatandaan nyo, yung dalawang F sabay na napoproduce. So, FADH, dun lalabas si fumarate. So, fumarate, FADH. Okay? And then finally nga, susu, gatas. So, remember, how many carbon dioxide? 2. How many NADH? 3. How many FADH? 1. How many GTP? 1. Anong purpose ng lahat ng yan? So, si acetyl-CoA, bakit kailangan pumasok sa Krebs cycle? Bakit kailangan sumakay ng taxing oxaloacetate? Kasi every time iikot siya, marami tayong mapoproduce na ATP. So, ito lang yung itsura ng molecules. So, this diagram just shows you the molecules and the number of carbons. Again, similarly, you will see where NADH and FADH are produced. Always remember, 2 carbon dioxide lang. Let's now go to the details of the individual steps. So first, acetyl-CoA combines with oxaloacetate to form citrate. The enzyme is known as citrate synthase. So nothing special kasi wala naman siyang dehydrogenase. And then citrate is converted to isocitrate. So the enzyme is known as aconitase. So aconitase is something you need to remember because aconitase can be inhibited. The inhibitor of aconitase is fluoroacetate. Fluoroacetate is found in plants, and so sometimes when grazing animals eat these plants, magsastop yung Krebs cycle nila. Hindi sila makakaproduce ng ATP. Also, some anti-cancer drugs and pesticides are metabolized to 5-fluoroacetate. To fluoroacetate, therefore, no, inhibiting this part of the Krebs cycle. So, ano mangyayari? Pag nabigay tayo ng fluoroacetate, what will accumulate? What will accumulate will be citrate. Kasi hindi nga natin ma-convert si citrate to isocitrate. Next, isocitrate is converted to alpha-ketoglutarate. So this is the rate-limiting step of Krebs cycle, isocitrate dehydrogenase. Dehydrogenase, it will produce NADH and carbon dioxide. That's carbon dioxide number 1. The next step is alpha-ketoglutarate to succinyl-CoA, catalyzed by alpha-ketoglutarate dehydrogenase. Ito, tatandaan nyo din to. Kasi si alpha-ketoglutarate dehydrogenase, pinsan niya si pyruvate dehydrogenase. At dahil magpinsan sila ay magkasing arte. No? Magkasing arte sila. So, what are the cofactors kaya of alpha-ketoglutarate dehydrogenase? Wag kayong magdaya. Hindi pwedeng B1, B2, B3, B5, no? So, kailangan kabisado nyo muna yung main names, no? So, that will be thiamine, FAD, NAD, coenzyme A, and lipoic acid. So once again, derived from B1, B2, B3, B5, and lipoic acid. So arsenic binds to which coenzyme? Lipoic acid. So pag may arsenic poisoning, hindi magtatrabaho si pyruvate dehydrogenase tsaka si alpha-ketoglutarate dehydrogenase. Anong sasabihin nila pareho? Ayoko nga. No, magkasing arte sila. So again, this will produce carbon dioxide and NADH. This is the second carbon dioxide molecule, the second NADH. The next step, succinyl-CoA to succinate, succinate thiokinase, susu, gatas. This step produces GTP, which is an ATP equivalent. In fact, it is su, no? Succinyl-CoA succinate substrate level phosphorylation. So yung substrate level phosphorylation, hindi lang sa glycolysis nangyayari. Nangyayari din siya sa Krebs cycle. So if they ask you which step in the Krebs cycle produces ATP via substrate level phosphorylation, it is this step, the succinate thiokinase step. Because GTP is an ATP equivalent. Succinate is converted to fumarate using succinate dehydrogenase. Succinate to fumarate, F. So therefore, this will produce FADH. Kung saan lalabas si F, fumarate, dun din lalabas si FADH. Then fumarate converted to malate, fumarase. Nothing special. And then malate is converted to oxaloacetate, malate dehydrogenase. Because it's a dehydrogenase, we will produce NADH. Bakit wala ng carbon dioxide? Bakit NADH na lang? Kasi dalawang carbon dioxide lang yung mapoproduce. Dalawa lang yung pumasok, dalawa lang yung lalabas. This diagram illustrates saan lumalabas yung carbon dioxide, NADH, FADH, and GTP. Remember here, GTP is an ATP equivalent. Substrate is acetyl-CoA. So if we now count how many ATPs are produced in the Krebs cycle. So when we convert, NADH, ilang NADH ba yung na-produce? 3. Each of those NADH molecules will be converted to 2.5 ATP. So 2.5 times 3, 7.5. How about FADH? 1 molecule, so 1 times 1.5, that's 1.5 total. And then GTP is equivalent to 1 ATP. So all in all, pag may pumasok, if there's 1 acetyl-CoA molecule that enters the Krebs cycle, we get 10 ATPs. So di ba ang dami, no? So every acetyl-CoA will get 10 ATPs. So if we go back to the very beginning from glucose, no? So if we begin from the start, starting at glucose, when you convert glucose to pyruvate, no? So aerobic metabolism tayo, may oxygen, may mitochondria. If you convert glucose to 2 molecules of pyruvate, you get 5 or 7 ATPs. Depending on the shuttle. So kapag malate aspartate, mas safe. So ilang ATPs total? 7. Pero pag glycerophosphate, naho-hold up tayo, ilang ATPs lang? 5. And then, the pyruvate will be converted to acetyl-CoA using the enzyme pyruvate dehydrogenase. So you must remember, no, when you convert pyruvate to acetyl-CoA, so as you can see here, you also produce NADH. And each of those NADH molecule, we can bring to the Krebs cycle. Kaya merong 2 NADH, no, times 2.5, that's 5. And then finally, bawat pyruvate molecule will convert each pyruvate to acetyl-CoA. Every acetyl-CoA you get 10. So 2 times 10, 20. So all in all, in aerobic conditions, during aerobic conditions, if you completely oxidize glucose, you will get how many ATPs? 30 or 32. Okay, so once again, do not forget this step, pyruvate to acetyl-CoA using pyruvate dehydrogenase. That will yield 2 NADH molecules. So in summary, glycolysis, 5 or 7 ATPs, glucose to pyruvate. And then, each pyruvate molecule will produce NADH. So 2.5, 2.5. And then, each acetyl-CoA will enter the Krebs cycle. And every Krebs cycle will produce 10. So pag tinotal natin lahat yan, it becomes 30 or 32. So is that clear? So compare that with anaerobic glycolysis, anaerobic metabolism. If you have anaerobic metabolism, how many ATPs lang ang makukuha natin from glucose? 2. Just 2. Why? Kasi 4 ATPs generated, 2 ATPs invested. Kahit nakakuha tayo ng NADH doon, hindi naman natin siya magagamit kasi wala nga tayong mitochondria or oxygen. Saan natin gagamitin yung NADH instead? In anaerobic glycolysis, recall, saan ginagamit ang NADH sa anaerobic glycolysis? You use it to convert pyruvate to lactate using the enzyme lactate dehydrogenase. So aside from generating ATP, there are other uses of the Krebs cycle. So number 1, citrate. Citrate will be used for fatty acid synthesis. So during the well-fed state, magki-Krebs cycle tayo, yung citrate, gagamitin natin yan to synthesize fats. So we will discuss that later dun sa lipid metabolism. Next, succinyl-CoA. Succinyl-CoA is used in heme synthesis. In the first step of heme synthesis, glycine combines with succinyl-CoA to form delta aminolevulinic acid. Delta aminolevulinic acid. So, glycine plus succinyl-CoA. So, tatandaan heme synthesis requires succinyl-CoA which combines with glycine, an amino acid. And then finally, malate. Malate is used for gluconeogenesis. So, the first step of gluconeogenesis, kailangan natin. So, dun sa first few steps ng gluconeogenesis, gagamitin natin to as I will show you later. So, remember the following. Number 1, the cycle does not synthesize new oxaloacetate. So, si oxaloacetate, yung taxi na sumundo kay acetyl-CoA, umiikot-ikot lang siya. Kung sino yung sumundo kanina, siya pa rin yun. Siya pa rin yun. There is no hormonal control. Walang effect sa kanya yung insulin and glucagon. Whether you're well-fed or fasting, it goes on and on and on. And then try to memorize what the enzymes and where you produce carbon dioxide, NADH, FADH, and GTP. We have this concept of anaplerotic reactions. Ano yung ibig sabihin ng anaplerotic reactions? Because yung Krebs cycle, di ba, kinukuha natin yung mga products niya. Pwedeng dalhin sa protein synthesis, pwedeng dalhin sa fatty acid synthesis, pwedeng dalhin sa heme synthesis, no? So, may mga intermediates na pwedeng maubos. Hindi pwede yung gano'n, no? Otherwise, titigil yung Krebs cycle. So, kailangan kung may kumukuha, meron laging nagre-replenish. So, right? The anaplerotic reactions ensure that the substrates, the intermediates of the Krebs cycle are always replenished. So what are the anaplerotic reactions? Tatandaan nyo lang to. The most important one is pyruvate carboxylase. So pyruvate carboxylase ensures that there is a constant supply of oxaloacetate. So other important anaplerotic substrates, glutamine, glutamate, aspartate, and propionyl-CoA. So you will see here, this one, pyruvate carboxylase. It ensures that there's constant supply of oxaloacetate. So you will see that, So using pyruvate carboxylase, it makes sure that you always have oxaloacetate in the Krebs cycle. What are the other anaplerotic reactions? Look here, glutamate ensures that there's always alpha-ketoglutarate using transamination. Aspartate, tinatransaminate naman natin to oxaloacetate. Okay? So, yun lang yung ibig sabihin ng anaplerotic reactions. These are the reactions that ensure there is that the Krebs cycle does not run out of intermediates. The next pathway we will discuss is gluconeogenesis. So, as I said, gluconeogenesis is the opposite of glycolysis. So, when we say gluconeogenesis, Genesis create, new glucose. So why do we need to create new glucose? So imagine niyo yung ganito, no? So for example, ikaw yung surgery intern, no? Ikaw yung surgery intern, tapos pinatawag ka sa OR, no? Pinatawag ka sa OR, mag-a-assist ka, no? Excited ka pa, akit ka sa OR. Kaya lang pagsabi sa'yo, no, ang i-assist mo pala Whipples, no? Whipple procedure. Tapos mano-mano lahat, wala pa ng mga ligature, wala pa ng mga staplers. So, and then first time gagawin ng surgeon. Okay? First time gagawin ng surgeon. So, nag-breakfast ka naman, no? So, umakyat ka sa OR, 6 o'clock ng umaga. Pag nag-breakfast ka, yun yung magiging source mo ng glucose, correct? No? Yun yung magiging source mo ng glucose. However, pagdating halimbawa ng 12 noon, di ka pa din kumakain, no? O kaya 6 p.m., hindi ka pa din kumakain. So remember, yung glucose na kinain mo kanina, 4 hours lang yung itatagal nun. So after 4 hours, wala ka ng glucose from the food that you ate. So hindi naman pwedeng hayaan na lang ng body natin na bumagsak yung glucose levels. So what happens is, after 4 hours, so at around 10 o'clock, your body will try to find new sources of glucose. And one of those pathways that replenish glucose during the fasting state is gluconeogenesis. So kailangan nating mag-create ng glucose. Kasi nga, hindi na tayo kumakain, no? Nandun na tayo sa fasting state. So, gluconeogenesis, it is the process of synthesizing glucose from non-carbohydrate precursors in order to prevent hypoglycemia during fasting. So, ano yung concepts dito? So, gluconeogenesis, kailangan mong gumawa ng glucose kasi hindi ka na kumakain. Pag hindi mo yung ginawa, what will happen? Without gluconeogenesis, you will become hypoglycemic. So where does it occur? It occurs in two organs, the liver and in the kidney. So si liver, 90% will happen in the liver. Kidney, 10%. During a prolonged fast, up to 40% can happen in the kidney. But really, the majority will happen in the liver. It occurs in both mitochondria and cytosol. So in biochemistry, there are 3 pathways that occur in both mitochondria and cytosol. No? Tatandaan nyo to. Ang mnemonic sa kanila, HUG. H-U-G. So remember, it takes 2 to hug. No? So laging mitochondria and cytosol. No? 2 to hug. No? It takes 2 people to hug. So H, heme synthesis. U, urea synthesis. And then G, gluconeogenesis. So remember, gluconeogenesis. So say it again, gluconeogenesis. Okay, that will help you remember, no? That gluconeogenesis happens in the liver and kidney. Again, gluconeogenesis. Gluconeogenesis. Liver and kidney. Okay, so liver and kidney. Gluconeogenesis. So what are the substrates? So sabi natin, gagawa tayo ng glucose from non-carbohydrate sources. So 4 possible substrates. Number 1, all intermediates of glycolysis and the Krebs cycle. Pwede natin gamitin. So basta intermediate ng glycolysis, basta intermediate ng Krebs cycle, pwede natin yung ibalik na glucose. So for example, ano ba yung mga intermediates na alala nyo? Oxaloacetate, okay, pyruvate, phosphoenol pyruvate, fumarate, malate, lahat yan. Basta nasa Krebs cycle, lahat. Basta nasa glycolysis, pwedeng maging glucose ulit. Number 2, lactate using the Cori cycle. I will show you this later. No? So lactate using the Cori cycle. Number 3, among lipids. Sa lipids, the glycerol and propionyl-CoA molecules from triacylglycerol. So we will discuss this later when we discuss triglycerides. So glycerol and propionyl-CoA, they can become glucose. And then finally, among proteins, the carbon skeletons of glucogenic amino acids. So always remember these 4. So sa carbohydrates, yung intermediates ng glycolysis and Krebs cycle, yung lactate from the Cori cycle. Number 3 naman sa lipids, yung glycerol and propionyl-CoA. And number 4, the carbon skeletons of glucogenic amino acids. So pag fasting tayo, yung mga yan ang pwede natin gawing glucose. Please memorize this. So sino kaya yung pinaka-important? The most important substrates during fasting for gluconeogenesis are your proteins, the amino acids. So this is the reason why, no? So ito yung reason kung bakit yung mga patients who are chronically ill, so those who have been admitted in the ICU for a long time, or those who have severe burns, ano nangyayari sa kanila? They lose their muscle mass. Why? Because their muscle mass, yung muscle nila, kino-convert to glucose. Now, if you do not give them enough nutrition, the muscle mass, their muscle, the proteins in the body will be catabolized and converted to glucose. Kasi mas kailangan natin yung glucose kesa yung muscle, yung protein. So, the product of gluconeogenesis is glucose. So, ang endpoint ito, gusto natin gumawa ng glucose kasi nga hindi tayo kumakain. The rate-limiting step is fructose 1,6-bisphosphatase, which converts fructose 1,6-bisphosphate to fructose 6-phosphate. So essentially, from 2 phosphate groups, tatanggalin natin yung isa para maging fructose 6-phosphate na lang siya ulit. So this is gluconeogenesis. As you can see, very similar siya to glycolysis, diba? Kanina lang pababa, ngayon naman pataas tayo. Okay, and if you remember again, kanina meron tayong mga enzymes na double arrow. Sila pa din yon. So glycolysis and gluconeogenesis, they share enzymes. Sino kaya yung exception? So glycolysis and gluconeogenesis, they share enzymes except for yung tatlong irreversible. The 3 important steps. So, do you remember the 3 important steps? Step numbers 1, 3, and 10. So, yung tatlong steps na yun, kailangan nating palitan. Kasi nga, irreversible sila. So, kung gusto nating umakyat, they need to be replaced by a different set of enzymes. Okay? So, ano na yung tatlo? Step number 1, glucokinase, hexokinase. Step number 3, phosphofructokinase 1. Step number 10, pyruvate kinase. So, sinong una natin kailangan i-reverse? No? So, kung gusto nating ibalik si pyruvate to glucose, we first need to reverse pyruvate kinase. So, you will see here, pyruvate kinase, this step is reversed by 2 enzymes. Pyruvate to oxaloacetate, which is catalyzed by pyruvate carboxylase. And then, oxaloacetate is converted to phosphoenolpyruvate using PEP carboxykinase. And then we will go up using the same enzymes as glycolysis. What's the next enzyme that needs to be reversed? PFK1. So kung kanina yung PFK1 nagdagdag ng phosphate, ngayon naman babalik na rin lang natin. Babawasan natin ng phosphate. So fructose 1,6-bisphosphatase, we remove 1 phosphate. Fructose 1,6-bisphosphate, tatanggalin natin ng isang phosphate using fructose 1,6-bisphosphatase, and we end up with the fructose 6-phosphate. So, kaya siya phosphatase. We remove phosphate. And then the last step, okay, glucose 6-phosphate is converted to glucose. So, tatanggalan ulit natin ng phosphate si glucose. From glucose 6-phosphate, magiging glucose na lang siya. And that's glucose 6-phosphatase. So, remember these 3 reversals, no? Yung tatlong yan, tinatanong kasi yan, no? So, for example, they might ask you, which of the following enzymes will reverse phosphofructokinase 1? And then they will give the list of enzymes. Okay? So, sasagot mo, fructose 1,6-bisphosphatase. O kaya, they will ask you, which of the following enzymes is present in both glycolysis and gluconeogenesis? So, ang sasagot mo, yung wala dyan, yung hindi reversal. O kaya tatanungin ka nila, sasagot natin, no? So, kung present siya in both, no? Di yung mga ibang-ibang enzymes. Pero kung nangyari, tinanong, which of the following enzymes is present only in glycolysis? No? So, ang sagot mo doon, 1, 3, or 10. O kaya, which of the following enzymes is present only in gluconeogenesis? Di ang sagot natin doon, either pyruvate carboxylase, PEP carboxykinase, fructose 1,6-bisphosphatase, or glucose 6-phosphatase. Is that clear? So, just remember, no? Kaya natin sila papalitan kasi nga irreversible yung sa glycolysis. So, let's look at the details, no? So, you will see here, pyruvate is converted to oxaloacetate. This is catalyzed by pyruvate carboxylase. So, tandaan nyo kanina, no? It's an anaplerotic reaction sa Krebs cycle. Most important anaplerotic reaction. Here's the thing you need to remember. Pyruvate to oxaloacetate catalyzed by pyruvate carboxylase. Pag narinig nyo yung word na carboxylase, automatic. The cofactor is biotin. So, there are 3 important carboxylation reactions. No? There are 3 important carboxylation reactions. Okay? So, you have, okay, there are three important carboxylation reactions. Number one, your pyruvate carboxylase step, which converts pyruvate to oxaloacetate. Number two, your acetyl-CoA carboxylase step, which converts acetyl-CoA to malonyl-CoA. And number three, your propionyl-CoA carboxylase step, which converts propionyl-CoA to methyl-malonyl-CoA. So, yung tatlong enzymes na yan, lahat yan kailangan nila ng biotin. No? So, remember those three steps. Pyruvate carboxylase, acetyl-CoA carboxylase, and propionyl-CoA carboxylase. Babalikan natin sila, no? So, when we discuss the specific pathways. And then, oxaloacetate will be converted to phosphoenol pyruvate. It's catalyzed by PEP carboxykinase. This one requires GTP. So, ito kailangan ng GTP. It's energy requiring. Next, reversal. Step 9. So this one, okay? So, this will reverse fructose 1,6-bisphosphate to fructose 6-phosphate. So, you will see, dahil ang nire-reverse niya, no, ang nire-reverse niya yung PFK1. PFK1 was the rate-limiting step of glycolysis. So, sa gluconeogenesis, yung fructose 1,6-bisphosphatase, siya din yung rate-limiting step. So, baligtaran lang yung dalawang rate-limiting step. Nire-reverse lang nila yung isa't isa. No? So, you will see here from 2 phosphate groups, tatanggalan natin ng isang phosphate na lang yung nakakabit. O, sino ngayon yung nakita nyo? Nakita nyo to? Si fructose 2,6-bisphosphate. Naalala nyo ba siya? So, remember, si fructose 2,6-bisphosphate, diba? Nandun siya kanina sa glycolysis. Anong role niya sa glycolysis? It's the most important activator of glycolysis, 'di ba? It activates PFK1, no? So saan nanggaling si fructose 2,6-bisphosphate? Pinroduce siya ni phosphofructokinase 2. So kung ina-activate niya si glycolysis, ano kayang gagawin niya kay gluconeogenesis? Eh di dapat i-inhibit. So you will see here fructose 2,6-bisphosphate actually has dual function. Not only does it activate glycolysis, it also inhibits gluconeogenesis. Okay? Finally, the last step is the conversion of glucose 6-phosphate to glucose. So you will see here, from glucose 6-phosphate, kino-convert natin siya to glucose. Now, I want to emphasize this. This step is very important. Bakit natin kailangang tanggalin, no? Bakit natin kailangang tanggalin yung glucose? Bakit natin kailangan tanggalin si phosphate? Ba't hindi pwedeng glucose 6-phosphate na lang? Okay? Bakit hindi pwede? Ganito kasi yun, no? So, when we look, okay, let me just do this. Okay. So, you will see, so kunyari ito yung liver cell. Okay? This is the liver. Okay? Ito si glucose. So remember, gluconeogenesis happens in the liver or in the kidney. So si glucose, pag may nakakabit siya na phosphate, masyado kasing malaki si phosphate. Hindi siya makakalabas, no? So the only way para makalabas ito dito, no, papunta sa other tissues, kailangan matanggal si phosphate. So kung hindi natin tatanggalin si phosphate, hindi makakalabas si glucose. Always remember that. Okay? So, always remember that. So, you see here, this is the importance of this step, the glucose phosphatase enzyme. We need to remove phosphate. If we don't remove phosphate, hindi makakalabas si glucose. This step is shared with glycogenolysis. So, yung glycogenolysis, ito din yung last step niya. The removal of phosphate to produce glucose. Okay? So, once again, ano lang ginagawa natin sa gluconeogenesis? Yung 4 na groups of molecules, no, yung in-enumerate natin kanina, ibabalik natin sila to glucose. I-reverse lang natin yung glycolysis. At para ma-reverse yung glycolysis, what do we need to do? We need to replace enzymes 1, 3, and 10 of glycolysis. Memorize the reversing enzymes. Okay. Anong importance ng removal of phosphate? You need to remove phosphate so that glucose can be given to the other tissues. So always remember this, anong goal ng gluconeogenesis? Sino nga mag-undergo ng gluconeogenesis? Which organs? Gluconeogenesis. gluconeogenesis. Liver and kidney. Okay, saan silang organelle? Sa cytosol, tsaka sa mitochondria. No? So, both cytosol and mitochondria. Anong bang goal? So, si liver, bakit siya mag-gluconeogenesis? Kasi gusto niyang mag-provide ng glucose to other tissues. Sasabihin niya, hindi na sila kumakain. No? Naku, hindi na ako kumakain. Wala nang glucose yung tissues. Ang gagawin ni liver, yung mga compounds na binanggit natin kanina, i-convert niya yun to glucose para merong magamit na glucose yung ibang tissues. At para makalabas si glucose, dapat matanggal natin yung phosphate dun sa final step. Okay? So, this enzyme, glucose 6-phosphatase, will do that. So, let's go to the Cori cycle. Sabi natin kanina, no, si Cori cycle, this is the pathway that converts lactic acid back to glucose. This is the pathway that converts lactic acid back to glucose. So, if you remember, no, so itong Cori cycle, love story to actually, no? So, love story to. Love story to ni liver, tsaka ni muscle. No? Love story ni liver, tsaka ni muscle. So, you will see here, si muscle, mahal na mahal yan ni liver. No? So, si muscle, mahal siya ni liver. Ito si muscle, actually, no? Alam naman niya, no? Alam naman niya na mahal siya ni liver. Pero si muscle, parang, okay, sige. Okay? So, now, one day, si muscle, nag-exercise siya. So, nag-exercise siya na matindi, yung glucose naging lactate. No? Kasi nga, nag-anaerobic glycolysis, no? Yung glucose naging lactate. Yung lactate, no? Useless yun sa kanya. Magkukos lang yan ng cramps. So, anong gagawin ni muscle? Si muscle, lalapit siya kay liver. Sasabihin niya kay liver, liver, nasobra na ako sa exercise. No, sobra na. Nag-produce ako ng lactate. Yung lactate ko, pwede mo bang gawing glucose ulit? So, anong gagawin, no? Anong sasabihin ni liver? Si liver, dahil mahal na mahal niya si muscle, sasabihin niya, sure, kahit ano, para sa'yo. No? So, anong gagawin ni liver? Yung lactate, kukunin niya. It will travel in the blood, papunta kay liver. And in the liver, what will happen? Lactate will undergo gluconeogenesis. So, lactate will be converted back to pyruvate using lactate dehydrogenase. And then, pyruvate will go up, mag-gluconeogenesis si liver, para yung glucose na ulit siya. Ano mangyayari sa glucose? Si liver, pupunta na siya kay muscle. Namin niya, muscle, ito na yung glucose na ni-request mo from me. Mahal mo na ba ako? No? Sasabihin naman ni muscle, hindi pa eh. No? Maybe next time. No? Maybe next time. So next time, nag-exercise ulit siya. No? Nag-exercise ulit siya. So yung glucose, naging lactate na naman. No? Naging lactate na naman. Ano na naman ang gagawin ni muscle? No? Gagawin na naman ni muscle, yung lactate, bibigyan niya kay liver. Liver, pwede mo ba itong gawing glucose for me? Ano sasabihin ni liver? Sure. Kahit ano, para sa'yo. So the lactate is again converted back to glucose. Okay? Using gluconeogenesis. Kaya nga diba, this, okay, lactate converted to glucose using gluconeogenesis. Si liver, may glucose na liver. Dadalhin niya yung glucose kay muscle. Muscle, ito na yung glucose na ni-request mo from me. Mahal mo na ba ako? Hindi pa eh. Maybe next time. No? So, paulit. The end. No? So yan yung love story ni liver tsaka si muscle. So yan yung Cori cycle. So tatandaan niyo lang, no, yan okay? So si liver, no, tsaka si muscle. Si muscle paasa, no? So si muscle paasa. So tapos si liver, no, ano si liver? Hindi naman tanga, no martyr naman, no. So si liver martyr, no, nagpapaka-martyr siya para kay muscle, no. So anong gagawin niya ulit? So the Cori cycle tells you how lactate in exercising muscle is converted back to glucose in the liver, and then it is returned to the muscle, no. So kailangan natin dyan is yung gluconeogenesis. Now, here's something you need to remember. Akala nyo ba madali lang mag-convert ng lactate to glucose, no? Or pyruvate to glucose? Hindi siya madali, no? In fact, to be able to create glucose from pyruvate, we need energy, no? So you will see here, look, Okay? To be able to create pyruvate, to be able to create glucose, you need, right? Look, you need two GTP and 2 ATPs. Okay? Another 2 ATPs here. No? So you will see 6 ATPs in all. No? So parang 6 ATP equivalents. 2 GTP, 4 na ATP. At the same time, we need to oxidize 2 NADH. Okay? So magastos. So para makagawa tayo ng isang glucose molecule, actually, si liver gumagastos pa siya. So where do we get that? Akala ko ba hindi na tayo kumakain? So kung hindi na tayo kumakain, saan tayo kukuha ng energy para mag-create ng glucose? This one. To be able to create glucose, we need to break down fat. So it's beta oxidation. So you need to beta oxidize fat so that we can create glucose during the fasting state. So tatandaan nyo yan, ha? So if they ask you, which pathway provides the energy for gluconeogenesis? The answer is beta oxidation. So you need to burn fat to create glucose. So some clinical correlates. So what is glucosuria? No? So glucosuria means, no, you have hyperglycemia more than the renal threshold. More if your blood glucose level exceeds 180 milligrams per deciliter, then your glucose will begin to spill into the blood. No, the glucose will begin to spill into the blood. Next, at the same time, no, so not into the blood but into the urine, no. So pag more than 180 milligrams per deciliter, kaya yung mga uncontrolled diabetics, di ba, kaya nilalanggam yung ihi nila kasi the glucose now spills into the blood. Critically ill patients, bakit yung mga patients kunyari na ICU or burn? Akala ko ba hindi sila kumakain? Bakit sila nag-hyperglycemia? So what happens during stressful situations, no? So there's excessive gluconeogenesis because of elevated levels of epinephrine, cortisol, no? So epinephrine, cortisol, ano nangyayari diyan, no? They stimulate gluconeogenesis. Ayaw natin 'yun, no? Kasi hyperglycemia is associated with poor outcome. So kaya, no? Kaya yung mga pasyente, no, na mayroong severe illness, if your patients have severe illness, dapat well replaced, no? So well nourished sila, otherwise mag-gluconeogenesis sila ng mag-gluconeogenesis, they will become hyperglycemic, right? And at the same time, dapat, no, kung kailangan nating i-control yung sugar nila, sometimes, no, kahit hindi sila diabetic, they are put on insulin. During pregnancy, no, there's high fetal glucose consumption. So that's why sometimes mothers become hypoglycemic, no? So there's a risk of fetal and maternal hypoglycemia, especially during fasting. And then finally, neonates. Yung mga neonates, di ba pag nag-duty kayo sa NICU, neonatal ICU, di ba nagsi-CBG kayo every so often? Bakit natin kailangan mag-CBG every so often sa mga neonates? Because yung mga neonates, number one, hindi pa well-developed yung enzymes nila for gluconeogenesis. So, hindi sila magaling mag-gluconeogenesis. And then number two, yung mga premature infants, especially those with low birth weights, kulang sila sa adipose tissue, no? So, dahil kulang sila sa adipose tissue, wala silang pang beta oxidation that will support gluconeogenesis. So, kaya ganun, mabilis sila mag-hypoglycemia kasi wala silang pang beta oxidize. And remember, beta oxidation provides the energy for gluconeogenesis. So, in summary, right? Biochemical pathways can be anabolic, catabolic, or both, no? So biochemical pathways can be anabolic, catabolic, or both. Anabolic build, catabolic destroy. What's the classic amphibolic pathway? The Krebs cycle. So it's not affected by insulin or glucagon. You must remember, meron tayong dalawang pinag-uusapan. Your fed state or the absorptive state, which is the first 4 hours after a meal. Anong mataas doon? Insulin. And the fasting state, which is 4 hours after a meal. Yun naman yung absorptive state. So ulit, fed state or absorptive, fasting or post-absorptive. So in the post-absorptive state or the fasting state, ano yun naman yung mataas? Glucagon. Second messenger of glucagon, cyclic AMP. So, tatandaan nyo lang yan. Papaulit-ulitin natin yan. Glycolysis is the major pathway for glucose metabolism in cells. It occurs in aerobic or anaerobic conditions regulated at 3 steps. So remember, first pair of opposite pathways, glycolysis and gluconeogenesis. Anong ginagawa natin sa glycolysis? Glycolysis is converted either to pyruvate or lactate. Pag may oxygen, may mitochondria, ang product ay pyruvate. Pag walang oxygen, walang mitochondria, ang product ay lactate. Remember the 3 important steps that are irreversible. Step numbers 1, 3, and 10. Remember the number of ATPs. Anaerobic glycolysis, just 2. Aerobic glycolysis, 5 or 7, depending on the shuttle. Okay? Next, remember that pyruvate, maraming pwedeng mangyari sa kanya. Pwedeng umakyat, bumaba, right or left. Pag anaerobic glycolysis pataas, pag aerobic glycolysis pababa, papuntang Krebs cycle. So pyruvate is converted to acetyl-CoA using pyruvate dehydrogenase. Remember the 5 cofactors. Bakit tayo pumapasok ng Krebs cycle? In the Krebs cycle, we will produce NADH, FADH, which is the greatest quantitative source of your ATP. We also get GTP, which is an ATP equivalent. At the same time, the 2 carbons of acetyl-CoA, it will be converted, oxidized to carbon dioxide, which we exhale. Okay, remember the important enzymes of the Krebs cycle. Gluconeogenesis prevents hypoglycemia during fasting by synthesizing glucose from non-carbohydrate precursors. Again, remember the 4 groups of biomolecules that can be converted to glucose. Number 1, all intermediates of Krebs cycle and glycolysis. Ano lang yung hindi pwede maging glucose? Acetyl-CoA. So remember this, acetyl-CoA can never become glucose. Once you convert pyruvate to acetyl-CoA, there is no turning back. Si acetyl-CoA pang Krebs cycle na lang talaga siya. Number 2, lactate. Lactate can become glucose using the Cori cycle. Remember the love story of liver and muscle. Number 3, among lipids, your glycerol and propionyl-CoA. And number 4, among proteins, your glucogenic amino acids. The most important will be your glucogenic amino acids. Sila yung pinaka-important na source of glucose for gluconeogenesis. Finally, glycolysis and gluconeogenesis are in opposite directions, reciprocally regulated by insulin and glucagon. So if I ask you, during the fed state, what will be active? Glycolysis. During the fasting state, what will be inhibited? Fasting, inhibited? Glycolysis. Kasi ano yung active? Gluconeogenesis. So isipin mo lang, no? Pag kumain ka, di marami kang glucose, no? Anong gagawin mo? I-metabolize mo ba si glucose or gagawa ka ba ng glucose? Siyempre, i-metabolize, no? Pag fasting ka, magme-metabolize ka ba ng glucose or gagawa ka ng glucose? Gagawa ka ng glucose kasi nga wala kang glucose. Okay? We will discuss more of this later. Thank you for listening.